from a set simulations.
Okay, thanks very much. So yeah, today I'm going to be talking about spectral simulation for MR spectroscopy. So just to give a very quick introduction, simulation of realistic MRS signals requires what we call the density matrix formalism, and it's derived from very basic first-year quantum mechanics, really. And there's a bunch of packages out there that kind of develop or use this same physics. There's a Gamma, which was written in C++. This was the first package, and since then there's been a lot of others. There's Spinach, which is a very powerful one, written in MATLAB. There's FIDE, which is a popular one used in the vivo community. There's JMRUI's NMR-scope-B, and there's a whole bunch more. I wrote one as well, which is called Mars, written in MATLAB, and you can even simulate it in the cloud these days, which I think is pretty cool. So, what do I mean by basis sets? So this is a fit. We've seen a bunch of fits, so we'll see a bunch more throughout these sessions. And what I mean by basis set is, how do we make these, which is kind of the spectral shapes that we then broaden and scale by amplitude in order to fit to our measured spectrum so that we can extract concentrations and whatever we want from there. And so one way to do that is you can measure them experimentally. That's not the purpose of this talk. This talk is how do we simulate them from quantum mechanics. So very quickly, I'm going to explain a little bit about my package. So it was a novel software package written from scratch, implemented in MATLAB, and you can run it. It's free to everybody, either via MATLAB executable or via Inspector's GUI. So these are the exact same physics under the hood, just two different ways to run it. And it can compute 23 metabolites in about 30 minutes for like a standard 3 pulse experiment like press or steam for 128 cube spatial points on a standard desktop. And I'll get into why that number of spatial points is kind of important. And so the reason why I wrote it is because, I wanted to do these large number of spatial points. It's quite important, especially for sequences like press. So I tried one package, NMR-scope-B at the time. This was around 2019. And it crashed with that number of spatial points. And then I got this excerpt from a paper which used Gamma. And they said the full 3D localized simulations demonstrated in this study utilized the power of multiprocessor systems and required several hours of computing time for metabolites with more than 4 spins. So this is 40 cube spatial points on a supercomputer. And so we did that in a lot less. And so, Mars can do that in a couple minutes, actually probably less than a minute. And some of these other packages that people use in the vivo community now can do that as well. So very quickly, I'm not going to have time to explain, the whole physics required to understand this. That would take probably several lectures, but I want to give just a little taste of how it works. So this is the density operator. Psi here is the wave function. Again, this is from quantum mechanics. And the time evolution of the density operator, which is referred to as the von Neumann equation, is given by this equation here. So H is the Hamiltonian operator. And so this is a matrix size of 2 to the m by 2 to the n. N is the number of coupled spins in this case. So for something like GABA, that would be 6. For glucose, that'd be 7. For an uncoupled spin, that would be 1. And so to give an analogy for people who are more familiar with the block classical vector formalism, this density operator, that's analogous to the magnetization. And then this von Neumann equation, that's analogous to the solution of the block equation. And so this is the Hamiltonian operator. And again, I don't have time to go through what all these terms, but I just want to explain kind of what they are. So, give a taste again. This is the Zeeman term. That's just due to the applied B0 field. You have the J coupling term. That's what gives us, all these nice multiplets that we measure. This is the gradient term. That's obviously controlled by the hardware. So it's, something we control. And then this is RF. Again, that's also controlled by the hardware. And then what do we measure? Well, once we know rho, we measure the signal is just given by this equation here. So TR is the trace, which is just the sum along the diagonal elements of this matrix. And IX and IY are the angular momentum operators. So if we know rho, then we can easily calculate S of T from this equation, and that's the simulated FID. And then, you can Fourier transform that. That's your basis sets that you end up using for fitting. And so really the question is, then how do we get rho? So I'm going to walk through how we do that just briefly through one particular pulse sequence. So this is press. And before we apply an RF pulse, we know the term rho is what's given by rho equilibrium. It's a very simple term. So, you know, you can look it up in a textbook. It's always the same. And then what we do is we obtain the time-dependent Hamiltonian from the pulse sequence. And so what I mean by that is, you know, if you walk through this in time, you have these time-varying gradient fields, you have these time-varying RF fields. And so you derive, you know, your Hamiltonian from that at each time point. And then it's just a matter of numerically solving this block equation step by step, basically. And one important thing is how you handle the coherence pathways. And so the way you do that in experimentally is usually you use gradients and sometimes phase cycling. But you probably don't want to do that in simulation because gradients are a little tricky. They interfere with something we call the 1D projection method, which I'll explain in a bit. Phase cycling is totally fine. The issue is though that, you know, if you need 128 phase cycling steps, then it takes 128 times longer because you have to simulate each of these phase cycling steps. And since computation is kind of interest, you know, computational efficiency, you probably don't want to do that. So the little trick we came up with, which is very simple, is the way phase cycling works is you just remove coherence order terms that you don't want. And so you can do this very easily mathematically just by going into the density operator and zeroing out the terms that correspond or that don't correspond to the desired coherence pathway. So for press, the coherence order pathway is -1, 1, -1, 1, -1. So all we have to do is zero out all the terms that don't correspond to that path. So, you know, at this point, it's -1. And just briefly, you know, you can also, the way we do this experimentally, like I said, is with crushers and phase cycling. So we have a little bit of work on that, how to do that experimentally. But yeah, you don't need to do that in simulation. And so then we go to, you know, the next term, that's 1, kill off all the terms that aren't 1. And then again, at the end, kill off all the terms that aren't -1. And so these are some validation that we got from Mars. So these are just, you know, some fits. And, you know, a fit doesn't necessarily validate a model for sure, but it is a very necessary but incomplete step that you need to do. And I really like this experiment that we got from some data that was laying around in the lab from measuring the T2 of glutathione, because they acquired, I believe, 8 different echo times with this editing experiment where you get these really distinct shapes. And so, you know, you can make sure that your produced spectral signatures look very similar to these kind of unique shapes. So I'm just showing 4 echo times, but we looked at all 8, obviously, and that's available in the paper there. And we applied this Mars to a whole bunch of other sequences. This is just one example of a fit obtained with a 20 millisecond S laser. And this one's what we call UT special, which is a 4 milliseconds UT special sequence. And so now I want to talk a little bit about computation. And so you might say, you know, why does it take so long? Like I gave you that example before, it was taking hours on a supercomputer to compute 4 coupled spins. And so I'll go through a little bit about that now. So computation is slow because we typically need to simulate something like at least 32 cubed spatial points. And so, you know, 32 cubed, that's 32,768. So naively, you might just say, well, let's simulate each of these spatial points individually. And that's why you end up getting these, you know, massive computation times. And the reason why you need to do all these different spatial points is that, you know, you don't just have one. When you're measuring a signal experimentally, you're not just measuring one in the center of the voxel. You're measuring the spatial profile that is kind of continuous from essentially 0 outside in the stop band into, you know, the nominal flip angle, and then back to 0 on the other side. And so there are 3 computational tricks that kind of I didn't develop them, but I developed one. We use them to kind of get this spatial time or this computation time down so much. One is called the 1D projection method. One is propagator for RF pulses, and the third is the coherence pathway selectors. And so going through that, you know, the computation time is slow, like I said, is 32 cubed spatial points. Matrix exponentiation takes about 2 milliseconds for a 128 by 128 density operator. So that's like, for example, an anomer of glucose. And then just to remind you guys, matrix exponentiation involves Taylor expansion of the matrix. So you got to do a whole bunch of these Taylor expansion. MATLAB does that for you, or Python or whatever. But that's why it's kind of a slow process. And then if you use 128 phase cycling steps to kill off your unwanted coherence pathways, And then there's about 2,000 time points in a typical pulse sequence. If you just multiply those all together for glucose, you get 194 days. So it's very slow. There's 2 nanometers of glucose, so that's a full year of computation. So obviously we don't do that, but yeah, that's what you would do from the brute force naive perspective. So the first trick that I think is really nice, and it's from this paper by Zhang, is basically you don't need to compute all n cubed spatial points. You only need to compute 3 times n spatial points. And this works for most sequences we use for in vivo spectroscopy, not all, but most of them. And I'm going to show this graphically. It's kind of something very obvious afterwards, but I don't think it was that obvious beforehand. So what the 1D projection method does basically is it says, well, sorry, this is an S-laser sequence. So for the first block here, you just have information in the X direction, right? I'm applying a gradient in the X direction. So why do I have to care about Y and Z? So you just ignore Y and Z for the time being. And then at the end of this X pulse, you add everything up, and then you kind of replicate your matrix along the next dimension, which in this case is the Y dimension. And now along this Y portion, you don't care about X, you don't care about Z, so it doesn't matter. You just do this one spatial dimension along Y, and then you do the final for Z. And so in this way, we're just simulating 3 times n spatial points. So, if it's 32, it's 3 times 32. You don't need to do each of those individually. And this is not like a computational trick. It's really mathematically the exact same thing. The second very useful trick is called coherence pathway selection. And so, like I said, gradients and phase cycling, they're techniques that just use to select a particular coherence pathway. But you can just zero these out mathematically in the density operator. And so this, can speed things up by whatever number of phase cycling you were using, which, is usually something like 128 for something like S-laser. And then the last is called a propagator for RF pulses. And it's also called the T matrix or transformation matrix. And so basically this is a way that you can calculate how the RF pulse transforms the pre-pulse to post-pulse density operator. And this is always the same for a particular RF pulse. So if you're always simulating the same sequence at the same field strength, you don't actually have to recalculate these density or these propagators. You can just do it once and then apply it. And then it just becomes a matrix multiplication. And this is really a lot of the computation time actually goes into making these propagators that transform from the pre-pulse to the post-pulse state. So you can really speed up computation. It's particularly useful for things like multi-TE studies, because in multi-TE you have the exact same pulses. And so you can get down simulations of these spectral basis functions to like seconds from basically 10, 20 minutes what they are otherwise. And so, like I said, these are not, these tricks are not approximations really. They're just computational tricks to speed things up. And so these are just some results of computation time for Mars. And so you'll see, for 128 cube spatial points for alpha glucose, we get 7 minutes. And, the calculation I gave before was 200 days. So, a very massive speed up. And other packages like FIDE, I think they use all these same tricks now. And so, when I came up with these results, I was very happy. I was like, well, so now we'll just all use these basis sets that use a large number of spatial points. And in our Mars paper, we kind of go into a little more why it's important to use a large number of spatial points. It really depends on the sequence and the field strength, but in general, you can't go too many. And so I said, that's it. Everybody's going to do that. And I got more pushback than I expected. Some people saying, well, I don't want to do that. Basically, can we just use 1 point in a hard pulse approximation? And, so I did a little bit of work and I thought, well, the answer is, I think no, but I guess we have to show that a little bit more. So this is one example of a simulation I performed with Mars for GABA, and this is a TE 30 milliseconds on, I believe, a GE press sequence. And LC model, that's not really, LC model doesn't simulate basis sets, but it's a basis set for LC model that I found on the internet. And I found the person who actually made this basis set, and they said, well, yeah, we used hard pulses. And so you can see these spectral shapes are quite different. And so to me, I wouldn't want to throw this in a fitting software because these are just, you can't scale these. These are just quite different spectra. And, we also did the simulation for the 3 different vendors. And again, you get quite different spectral signatures. But now the question is, this is like obviously a very sharp line width. We don't get this in vivo. It's very broadened out. What happens when we go to a more realistic scenario? And so what we did here is we synthesized this data. So this is like synthetic data, and we know ground truth everything. We know the concentrations, line widths, everything. And we created this using a GE basis, which corresponds to the GE press. And then we fit it using a hard pulse with the Siemens basis, with the Phillips basis, just to see what happens. And I'd say that, I think these fits are quite nice, regardless of the basis set. You do obviously get some variations. But, would this be lost in the noise in vivo? I think probably. So I don't think anybody would be too unhappy with these. But if you now look at the percent deviations of the concentrations, they're massive. So I highlighted a few here. Even for NAA, you get something like 4% for the hard pulse. And you might say, well, we have CRLBs. These are not CRLBs though. Like the CRLBs in this case are 0. This is noiseless data. This is, systematic deviation from the ground truth. And for these smaller metabolites, I mean, they're way off, right? For GABA1, you get 156%, which probably isn't that surprising because I don't know if we can really quantify GABA in these short TE spectra, but even NA or choline plus GPC, this is 10%, probably much higher than your CRLBs. And so we did some very similar work, or a colleague of mine did some very similar work in vivo, where instead we use experimental data. In this case, you don't know the ground truth, obviously, because we don't know ground truth concentrations, but we use the ground truth as essentially the values which have the proper basis set that matches the experimental data. So we have, I believe this is GE data, and so then we used a GE matched basis set would be the ground truth, and then we saw how much it deviates. And this was using LC model, and it tells a similar story. It's not quite as extreme, I think probably because of the inclusion of the baseline kind of smears this out, but you still get values, NAA down here, they range from 2, 5%, GLX, you get almost 20%, probably quite a bit larger than CRLBs. So this just really goes to show how important having a matched basis set is. And so very quickly, I'd like to explain how to use MARS just to try and increase adoption of this type of stuff so that nobody really has to use these basis sets they found in the internet unless they're 100% sure it matches their particular sequence. So there's 2 ways you can download them from the 2 links here. I'll go through really quickly how to do it in the inspector GUI because it's just, I think, really easy. So all you have to do, you select your sequence and your vendor, and this is for the stock, steam, and press sequences. We also have our implementation of S-Laser, which you can also get and put on your, I believe this is for Siemens. And then you just choose what particular metabolites you want. And you can edit this too if you think that our J coupling values are a little off or somebody comes out with new ones, or you want to add a new metabolite. It's very easy. It's just like a .mat file that you can edit. And then, these are standard parameters that should obviously match your experiment, things like your field strength, your receive bandwidth, number of points, echo time. And then you run the simulation, and then it will just spit out a directory that contains the .raw that you can then use to make your LC model basis set. And so, just looking at these metabolites, this is what it looks like. Here's one for lactate, just looking at a single metabolite. So this is obviously an echo time of 144 milliseconds. So very easy. So in summary, density matrix simulations are necessary, not necessarily, but you could always measure them experimentally, but it's kind of what most people do these days. And it's also, I think, really useful for simulating experiments. It can be easily achieved for stock sequences and S-Laser through the inspector GUI. And it can be run for any custom sequence. So, really anything. And if there's something that you think isn't implemented into MARS, then just email me. And I'd be happy to work with you to get that in there. And there's numerous high quality and well-maintained simulation packages available. So MARS, obviously I'm biased. I like that one because I wrote it and I know everything that goes into it. I think FIDE is also quite popular. And so I think, no one should really be using out of the box basis sets anymore unless they're really certain that it matches their exact sequence, which probably means you made it yourself or the person who made it. And if you need assistance simulating basis sets, please email me. And so I just want to talk a little bit that I think actually using a simulation, I talked this whole time how you simulate basis sets. I think it's really useful for things beyond that, actually. Because you can now just simulate an experiment that in some ways looks very realistic. And so I'll give an example of something I spent time on. I tried to develop an MQF S-Laser sequence with my good friend Dikoma Shungu for GABA. And in the end, it didn't work due to low yield. And it saved many hours in scan time. So, I was running a computer, desktop, basically free. I would say I probably spent about 100 hours. If you wanted to implement this on like a scanner, I think anyone who's implemented a novel sequence will tell you 100 hours is very conservative. At $600 an hour, that's $60,000 of savings. And you can do a whole lot more. You can do, we've investigated how CRLBs are related to standard deviations through simulation. You can optimize your choice of echo times through this type of experiment. We've done work on that as well. And then other people have investigated how metabolite concentrations correlate with the more, essentially the spectral broadening, the more overlap there is in your spectra. Relaxation we've looked into a little bit as well. And we've done some simulation as well with this using water suppression, and we coined that constrained optimized water suppression, or COWS. And so, I do want to end with saying experiments are still critical. Validation is very important. So simulations are more to guide experiments more than necessarily to replace them. And so with that, I'd like to thank everybody, especially my collaborators.
Any questions?
This was really, really nice. I enjoyed that a lot. Thank you. I just sort of had a question. I think your comment at the end, basically that we need to simulate the exact sequence that we're running is probably a little harder than I would go on that one. And I think for the average user, it can be very difficult to know what the scanner is doing. And on some vendors, for example, the pulse durations even can change based on calibrations. And so it actually can be the case that the simulation would change for each subject based on the sort of the pre-scan steps. And so I 100% agree with you, obviously, that we should be simulating stuff, but I think getting it exactly right every time is probably further than any of us really want to go.
Right. Yeah. I mean, I think that can be valid, and it depends on the sequence. When I said, being harsh, that's something for like press, where it's quite sensitive. For something like laser, it's far less sensitive. The higher field strength you go, usually these effects get exacerbated. So higher field strength people have to be more conscious of this than low field. I'm not aware, so I don't know. I have most experience on GE, which does not do that, does not change, like, for example, press or steam. They don't change with.
Then I should maybe be careful what I say. My understanding is that GE does or did stretch pulses based on things, but if I'm wrong, then I can retract that.
Yes, well, we talked to Ralph. I'm not aware of that for sure. There are ways to swap out the pulses. I would still say, though, let's say it did happen. If you keep a log of this, it's very easy to create a basis set for, okay, this time it did that duration, and like these simulations take 30 minutes. And if you have a dozen subjects, it's very doable. Now, if you had thousands of subjects, that would start to get intractable because it'd be a logistics nightmare. But I think it's still doable. But you're right. I mean, we're never actually encoding everything in there. But my position is you should do as good as you can, right? And I think it's reasonable at this point. Yeah.
I just wondered if you had any thoughts on how we enable people who don't have the ability to look at sequence simulations and that to be able to use these tools, because as a analysis software sort of developer, the primary question I get asked is, how do I make a basis set? And the answer is, well,
could you speak to the microphone? People can
sorry,
hear.
the primary question I get asked as some sort of analysis software developer is how do you make a basis set? And
right.
You've solved one bit with the, physically how do you run the computation, but how do we get people who don't have access to simulations, etc., to be able to use tools like Mars?
Right. I mean, yeah, so that's a good question. So Mars, like I said, you can download it, you can run it. There is a user guide. Obviously, for somebody who has no experience, it's like a little daunting to walk into it. Basically, Mars encodes all the information that's in the experiment. And so you're right, you do need to get access to that. So that means you need to get the RF pulse waveforms, and you need to get the gradients are actually a little easier, so that's probably not a big deal. Really, it's getting the RF pulses and getting the timings that are the two hard parts. I don't have a good answer for how to do that. I mean, that's something that people need to have experience on the scanner. And then if you make your own sequences, you know all that stuff. Like that's super easy. For something like stock, if you're just using a stock sequence like GE or Siemens or Philips, we do have those stock sequences in Mars already. So you can just choose that template. You just have to change the echo time. So that makes it a lot easier. But yes, I mean, probably contact me. It's a hard question for sure.
I would like to ask a question. Is it correct to assume that if you use ultra short echo time for your sequence, that the dependence on parameter, how many spatial points you have, or that is your sequence of basically a simulation start to be independent of those parameters for ultra short echo?
Yeah, that would be true. I mean, if you had an echo time of 0, then then that's true. As you get a longer echo, these effects do become more apparent, more or less. I mean, it can oscillate actually, depending. Like everybody knows the classical lactate at 144.
Yeah. So well, basically, you can forget about J evolution, and then I assume basically that you can assume also hard pulses instead of real pulses?
Yeah, I mean, that's something I would think, yes, that probably would be fine. It's something that I would test experimentally just to make sure. But yeah, my intuition is you're right that it probably doesn't matter for very short echo time.
You know, also relative to the first question, that if your simulation should be as close as possible to real experiment, but it is much more important for longer echo times.
It is definitely more important for longer echo time, but like I said, it oscillates. Like for lactate, it's more important at 144 than 288. So it's not this monotonic relationship. But for at 0, I would think, yeah, it probably doesn't matter at all, probably. Yeah, that's true.
Thank you.
But most people are not doing ultra short echo time stuff, right? So I saw the poster, but.
All right, let's move on to the next talk. Thank you. The next talk is from Anke Henning from University of Texas Southwest Medical Center. Talk title is a parametric fitting.
Thanks to the organizers to invite me to give this talk. And we will take the basis sets that we just saw a step further and really incorporate it into a parametric fitting software. And I will mainly explain how these fitting softwares really work. So a little bit more on the algorithmic side. So first of all, we have in spectroscopy a wealth of information. In proton spectroscopy, we can distinguish about 20 metabolites and compounds, but they're all overlapping with each other. And spectral fitting is the essential step to discriminate the different metabolites, contributing to such a kind of crowded spectrum. And most of the examples that I show in this talk will be proton, but I just wanted to mention that there are some other nuclei that are hardly considered in a lot of softwares that are really worthwhile to look at. And the basic principles that I talk about are also applicable to those. So the first, maybe before we really dive into the algorithmic side of spectral fitting, I want to stress what you heard probably yesterday already. Tune your MRS acquisition as good as possible, and then also your pre-processing steps, because if you have good data and feed it into the spectral fitting software, you will get reasonably reliable fits out of it. While if you have probably data which have major artifacts or even minor things like the phase is not adjusted properly or the eddy current correction is not done properly, let's say the fit is less reliable and you will get much higher standard deviation. So spectral fitting is just one step out of a lot. of other steps, and it cannot function reliably if the other steps, especially the acquisition and the pre-processing, is done correctly. And we saw in the previous talk that, of course, an appropriate data set that really reflects your experiment is of utmost importance. So now, what is spectral fitting and why do we do this? So we have to understand that the amplitude of the FYD in the time domain signal and the area under the peak in the frequency domain signal is correlated to the concentration of metabolite. And that's where we want to actually measure either the amplitude of the FID, which is the very first point, or the area underneath the peak. And you can do this fitting in the time domain. So you can really fit the FID or the echo signal directly, or you can do the Fourier transformation and have all these different spectral peaks. And you can do the fit in the frequency domain. There are different packages using either time domain fitting or frequency domain fitting, or even a combination of both. Then some packages don't really rely on basis sets, and they're more simplistic, and some of them are really applicable to especially non-proton MR spectra. And for instance, MRS is the most popular example, and it really fits only single resonance lines. So this is okay for spectra that are not very crowded, and if you are interested in just maybe a couple of lines. But the state-of-the-art approach today, which I would really recommend, is to fit a linear combination of complex spectral pattern, which is, for instance, what LC model is doing, but also a lot of newly occurring packages. And then what is the difference between time domain fitting and frequency domain fitting? So in the time domain, during the fit procedure, there's no transformation applied to the spectral basis set and data, while in the frequency domain fitting, most of the packages define the spectral model in the time domain, but then do a Fourier transformation of the spectral model as well as you provide the data as Fourier transformed. But it is really the spectral basis set that is Fourier transformed during the fitting step. So that is a bit more computationally expensive. Time domain fitting, it is much easier, especially to define line shape models. And frequency domain fitting has the big advantage that you can really select the frequency range for spectral fitting. So you can exclude large nuisance signals like water and lipids from the fit, which makes the convergence also easier. Uneven sampling can be accounted for in time domain fitting. That is, for instance, interesting if you have echo time series or you want to have really series of parameters that you don't want to equally sample. It doesn't matter so much if you have just in one D spectrum. And missing data points, for instance, at the beginning, or also some additional data points if you do maximum echo sampling or you do pulse acquire where some data points are missing, are not such of a problem. While in the frequency, you need to consider this one way or the other in the spectral basis set. The majority of the fitting packages combine the advantages of both and define the model actually in the time domain, but then do frequency domain fitting. But there are also some packages that really stick purely to the time domain. So then maybe what kind of parameters do we need to fit if we look into time domain versus frequency domain fitting? So we need, of course, to know where the peak is. Frequency, we need the relaxation rate, and that translates into a line width in the frequency domain. We need the amplitude, as I said, which is correlating with the concentration, and we need the area of the Lorentzian or Vogt line or more complex line shapes in the frequency domain, and the phase and some line shape models. So this is the major parameters that are fit, and we are mainly interested in the amplitude, but for reliable modeling, we definitely need to consider all the other parameters as well. Then I stole a table from Robin de Graaf's book that shows a little bit more in detail how these models look like. And so we have, for instance, here, the possibility of choosing different line shape models. If we stick to the parametric ones, I go to the non-parametric line shape models in a minute. So the most common ones are Lorentzian line shape models that really accommodates mainly the T2 relaxation effects, and Gaussian line shape models that consider the T2 star relaxation, which is related to the macroscopic and microscopic inhomogeneities, so static field inhomogeneities. And usually people eventually fit forked lines, which is a combination of T2 relaxation and T2 star relaxation, and most more modern software packages include also this. Then, yeah, we have the amplitude, we have the frequency phase, and here the T2 relaxation time is fit directly in the time domain, while the line width is really fit in the frequency domain. So then how is this information integrated into an actual spectral model and into a cost function? So let's look at, maybe I picked 3 packages. So 2 of them, the most used one, GMRUI, is a purely time domain fitting software. And the first expression here is from the MRS algorithm. And you can see that it really fits a linear combination of the amplitude, and it has here the line shape parameters, it has the frequency, it has the phase. And then it does simply a least square minimization. So this is really the basic step of spectral fitting here. And everything else is really trying to tune the model to accommodate, for instance, imperfections. So this book chapter here that I cited summarizes all the time domain fitting that is implemented in GMRUI very neatly. And it is important to say that this is the metabolic or the parametric modeling part, but a lot of softwares also have a non-parametric part, and then also consider some noise in order to model realistic spectra. In LC model, which is a frequency domain fitting package, we see similar things. We see first order and 0 order and first order phase. We see the concentrations, we see line shape parameters, we see frequency offsets. So it is really the same expression. And the difference here is LC model really, way it transforms the basis set and then executes the fitting in the frequency domain. And I want to illustrate something that is maybe important. So LC model uses a lot of regularization and also constraints in addition to the basic model, the basic minimization, in order to make the fit more stable. So these are taken from the original publication from Stephen Provence. So I did not look, I think recently the code became available. I didn't really look into the code to see whether this is what he's still doing, whether the software evolved, but this is what he described originally. And he has 2 regularization terms, these 2 here, that imply smoothness of his line shape model and the baseline, also the spline baseline model. And then he also defines probabilities of the chemical shift and line widths that match the realistic range that we find in vivo. And he kind of regularizes that in order to prevent deviation. And so all of these regularization really makes sure that none of these parameters deviates too much from actually realistically occurring situations in the spectra. But it also kind of enforces, of course, certain solutions, so which is not always good, and we will come back to this later. In addition, he also uses constraints. And so he says, okay, concentrations have to be larger or equals to 0. They cannot be negative, which makes sense. And then he imposes also constraints on the line width. It has to be also larger than 0, and also on the line shape parameters. So this is a way to make your algorithm converge more robustly. But it has also a little bit of the danger if you look or use a lot of regularization. It can also really force solutions that are maybe very reproducible but not accurate. So we come back to that. We, through our own software package on the market, which is Profit 1D very recently, and we made it available. I want to just illustrate on our example that you can do another choice in your fitting package. The choice is how to group parameters. Some of the parameters can be global, so it's the same for all the metabolites, which really reduces the ambiguity and the degrees of freedom. So if you say, okay, the entire spectrum has a global 0 order phase, or the Gaussian line broadening was really related to the microscopic and macroscopic, so it should be the same for all the metabolites, then you can simplify the algorithm by just grouping them into one global parameter instead of having metabolite-specific parameters, like, for instance, I think it was done in MRS. We can have metabolite or moiety, even moiety-specific parameters. Of course, we have to have it for the concentration. The T2 relaxation time obviously differs, and the frequency shifts. Of course, the frequency differs. So these has to be metabolite or even moiety specific and cannot be global. So this increases, of course, the degrees of freedom. And then we have here some global chemical shift of the entire spectrum again. So this is really, you have to make a choice that makes sense, actually, and it will help the convergence as well. And then I want to maybe point out, in our program, we only use the baseline smoothness regularization, not so much other regularization. But we played, or Tamas Borbert, he was a student, and in my group, he played around with different cost functions. And he tried to see, okay, what is the difference between pure frequency domain fitting, or then he did fit both. He did do a time domain fitting and the frequency domain fitting, and really looked at the errors that are, or the deviation from both models simultaneously, and let the algorithm guide by both inputs. So this is true TD-FD fitting, so time domain, frequency domain fitting. And then he also thought, well, our spectra are sparse, so we just need to look into the regions where we actually expect spectral peaks, and we don't necessarily need to put so much emphasis in the noise. And he tried that. So what we found is that it gets a bit better if your cost function gets a bit more complicated, but it is also not, it is maybe it can be too complex without too much gain as well. So all the details are summarized in this paper. So then once you defined your cost function and made reasonable choices about the parameters you want to fit and which are global and which are local and what kind of regularization you want, you have to still define what kind of optimization algorithm are you using. And the original LC model software, and then also Profit, uses the VAPRO approach. And that really distinguishes or separates the concentration fit, which is just a linear least squares fit, from a non-linear fit that is really fitting all the other parameters. So that can be done. It's not done by all packages, but it speeds up the process. Then you can implement iterations. So I'm not sure LC model is doing. I need to actually look into that code. But for instance, our package is doing it. And we increase the number of parameters and also the number of metabolites that we fit throughout the iterations. And especially in later iterations, we introduce more complex baseline and line shape models. And in the very first iterations, we only fit the main metabolites. I believe that LC model is doing that too. I think it fits also the main metabolites, main 3 or 4 metabolites first, and then goes on in another iteration. But I really have to look into the code to really find out what it exactly doing. It's not really described very well in the paper. So this can help you to guide the algorithm towards a sensible solution, and then also to converge. And you can also change constraints throughout the different iterations, so to make them tighter, for instance. Then solvers, the majority of the programs that I found use gradient descent methods. So Levenberg-Marquardt was actively used active set, linear and quadratic programming. But there are also packages that use just the pseudo-inverse. And then there are 1 or 2 packages using Bayesian methods. So altogether, spectral fitting, similar to what we heard yesterday, is an inverse problem. And all these solvers are appropriate, but there is no, to my knowledge, no systematic investigation on which of the solvers converges more robustly, which is, of course, of concern. So now we looked at this. So the next step is maybe to look at the non-parametric parts. So in the parametric model, we really put in all the prior knowledge that we have about the spectrum, including the basis set. But then there are some experimental imperfections, like whatever, eddy currents or other forms of line shape distortion. And that was considered an LC model. So LC model considers a eddy current kind of line shape model, which was apparently maybe in the early 1990s more of a problem than it is today. But also the JMRUI packaged access includes line shape models, and they investigated delta peaks and also spline line shape models. And what you can see here is that a normal Gauss model would not include these dip, and you would have these residues here. But if you have a complex and non-parametric line shape model, that can be accommodated, and the residue is much smoother. The disadvantage of these complex line shape models is a little bit, they of course influence your area underneath the peak estimate, and that can lead to some normalization issues. So all the fit may look perfect and the modeling is done essentially correctly, but just because of the introduction of these non-parametric line shapes, there is kind of a fudge factor that scales the spectrum a little bit up and down, and that is not usually considered in the further quantification process, and it makes it a bit difficult. I don't have a good solution to it, but it's happening, and it may explain some differences between different packages. Then I told you that you should always face your spectrum in advance. So if you have a spectrum that has a definite echo time, I mean, really do the phase correction properly. But there may be some exceptions, and I would always recommend against letting the fit algorithm do the phase correction. What I would recommend, if you know that your basis set or your data have some phase, and you did not correct it in the pre-processing step, which can be done by linear phase correction, by missing point predictions or so, then you can also simulate basis sets that just include the phase. So you know how many points are truncated, for instance, at the beginning of your data set in an either phosphorus ISIS experiment or pulsify experiment, or in a proton FID MRSI, this happens. And if you consider it in the basis set, then the spectral fitting software doesn't need to do the correction, and that's really much more robust. Spline baseline, that is a common issue. So this is an example of bad water suppression, and you have these spline baselines. So not all, but most packages include some spline baseline to model any experimental imperfection mainly. So again, the better your data are, the less of this spline baseline modeling is needed, the better will be your fit results, because you can imagine that, I mean, the spline can be adjusted in a different way, and it will definitely influence the area under your peak estimates. And we did with LC model a systematic comparison of different line or spline baseline stiffnesses. So they have a parameter that is called DKN-TMN. And if you set it to very high numbers, like 1, then it's very stiff, and to low numbers, it's very flexible. So the parameter really defines the number of knots in the spline. And what we generally found is that I mean, the residues look similar. So that is also, I mean, you can see, you can model the same spectrum really in different ways. But what we found from a quantitative analysis is the quantification results really differ between the settings. So we see here, for instance, really statistically significant differences in the estimates of multiple metabolites just based on the choice of the line, or the spline baseline stiffness. And then another parameter that is, of course, important is the macromolecule content, especially in short TE spectra. It is really also of utmost importance to have a reliable macromolecular model that really fits your data. And don't rely, for instance, on the implemented LC model versions. This is the bottom here in this graph. Yeah, it models some lines at the frequency offsets, but it really does not resemble the actual appearance of a experimentally acquired macromolecular baseline spectrum, which is generally recommended, I think, especially for shorter TE spectra. So this is also kind of an unsolved problem. The spline baseline, especially, and the macromolecules influence the estimates of the metabolite concentrations enormously. So what we did is we picked up an idea of Martin Wilson, and he said, let's not choose, the user shouldn't choose the number of knots in the spline baseline, but for each spectrum, and depending on how much of a line shape distortions you have, and we have here different simulated cases with different line shape distortions, there is an optimum of the number of knots, and you can really find it out in an automatic fashion. So in our software, it is adapted to the actual needs of the spectra itself, which makes it more user independent. What we also found, so we use less regularization in comparison to LC model, and we get slightly worse precision, which is really the reproducibility, but we get much better in the accuracy. So we simulated bases or test spectra. We added actual line shapes that were extracted from, or imperfect line shapes that were extracted from actual data. We added noise, we added different baselines. And what we find is that with our less regularized like fitting algorithm, we get closer to the input concentration that we know with simulated spectra, and LC model has a higher reproducibility, but really the accuracy is lower. And so this is also of concern. So you have to always balance accuracy versus precision. And the more you constrain and the more you regularize, the more you enforce a specific solution. And that might end up in high reproducibility, but it doesn't tell you you are accurate at all. So this is a major concern. Then some of the, so there are some specific situations which I will go over very quickly. So if you want to fit some unusual spectra, most fitting packages cannot really accommodate that. So one example is amino acids. So we try to fit macromolecular spectra just to quantify the macromolecules itself. And for that, we extracted histograms from the NMR structural biology database. And even the molecules or the amino acids are the same. If they're bound to different proteins, they have different chemical shifts. And this leads to such histograms. And these histograms have, of course, they are representing eventually the line shape of our macromolecules, but they have nothing to do with the full line shape. And so the fitting package like LC model cannot really accommodate that, for instance, because it assumes certain parametric line shape models and cannot really accommodate anything that deviates largely from it. So this is an attempt to fit a macromolecular spectrum with these extracted basis sets for the amino acids. And I mean, it matches quite okay, but it's clearly seen that the line shapes don't match. And so this is really an essential problem that most fitting softwares do not consider. Lipid is another thing. You need very good lipid models, for instance, for tumor spectra, also for muscle or heart spectra. And here I have to say, among the packages I have seen LC model, Stephen Provencher spent a lot of time to develop sophisticated lipid models that really even include things like intra and extramyocellular lipid in the muscle. And they work very nicely, and almost no other fitting package can actually do that. The last couple of minutes I want to spend on MRSI fitting. And there are some specific considerations because we have, of course, we fit in a large number of voxels in MRSI. They are not entirely unrelated. And you may use spatial prior knowledge in order to improve MRSI fitting results. And this is an example of a self-written MRS implementation applied to just even long echo time spectroscopic imaging from the 3 Tesla. And then in MRS application or implementation that also uses spatial priors. And the spatial priors did nothing else than assuming that the phases, the line shapes, and the frequency offsets jump. They don't jump between neighboring voxels, but the transition is smooth. This is the entire assumption. There was no assumption on the concentrations between neighboring voxels. And you see that the maps occur much smoother, only considering that there's probably a gradual transient. Another note maybe on MRSI is the macromolecular model. Like in single voxel spectroscopy, usually you have sequence parameters that you can adjust in a way in order to acquire experimental macromolecular models that have the exact same parameters. But in some circumstance, and this example is FID MRSI, the repetition time is very short, and it is actually not possible to acquire matching macromolecular models because of SAR constraints if you add double inversion sequences. And so we came up with a relaxation corrected macromolecular model that is extracted from experimental models in, for instance, semi-laser, but then corrected for differences in the relaxation time across field strengths, but then also across the different sequences. And with this setup, you can adjust your macromolecule to your parameters that you used in your spectral acquisition. And you get much better results. And if you use a wrong macromolecular model in fitting single voxel spectra, but especially also MRSI, your maps may really look a lot different. So the first row here shows, again, the LC model simulated macromolecular baseline, which doesn't perform well, and some of the metabolites are just fitted by it completely. So we don't see NAD or glutamine. The experimental macromolecular baseline that was acquired with semi-laser, alters, let's say, the appearance of the maps. And the simulated one really leads to a clear distinction, for instance, of NAG or glutamine, and also correction of the amplitude estimate of myoinositol. So this is also an influence parameter that, let's say, influences your results more than you may be aware of. So just consider this. Theresia, a student in my group, did a more systematic investigation of lipids, so lipid or no lipid suppression retrospectively, and then the choice of the spline baseline, flexibility, and the macromolecular model. And what she found is that the main influence factor is really the spline baseline, so that really changes the number of voxels that are fitted and the CRLB estimates, as well as also the reproducibility. So we did the test-retest study, and the spline baseline model was really the parameter that had the most quantitative influence in FYD MRSI spectra. Also lipid regularization is needed. Okay, so these are then data that consider all of this. And so with proper fitting, you can get reasonably nice spectroscopic imaging out of a large version. I will, in the interest of time, I skip this. Yeah. I know.
You finished.
Can I have 1 more minute or 1 or 2 more minutes to finish? I want to highlight the comparison of fitting software and the accuracy at the end of this talk. So in a book chapter that is recently published, I tried to summarize all the different fitting packages, and you will see how different they are with respect to the choice of optimization algorithm, iteration blocks, then whether they are accommodating multidimensional fitting, and then also really in the bounds and the regularization choices, and also whether or not they handle macromolecules or they can include a non-parametric baseline. There's really a large variation of different combinations of different options there. And with that, it doesn't, I mean, it's not let's say, cumbersome to also interpret the results of the recent spectral fitting challenge that showed that fitting packages actually perform quite different, and it also really depends on the input signal, for instance, Lorentz versus Gauss baseline or the SNR, and that is really explained by the different configurations of the different algorithms. I want to just finish off with the precision and accuracy. So there are, of course, the Cramer-Rao lower bounds that you can estimate, and that is just one estimate, and that is really a lower bound. So it tells you what is the minimum error. And I would also really always recommend to look at the covariance matrix that can be also derived out of the same Fisher information matrix. And you will see spectral overlap, and it identifies really from correlation coefficients which of the metabolites at your spectral line width and field strengths you can distinguish and which you cannot distinguish. So this is a worthwhile analysis. And then again, look at accuracy and precision, and not only at precision. So a lot of studies just look at precision, but it is really essential to validate fit packages against prior knowledge, and this can only be based on simulated data. So you need to evaluate accuracy. And with that, I go and show the cow. This is an important thing here. Just wanted to point out we can easily compete with Switzerland and Texas. We have 10.9 million cows running around. And a remark that we also have open positions in Texas.
Thank you. Probably for the sake of time, probably no time for a question for now. We can ask her later.
So we can move on to the third and last educational talk, which is entitled Quantification, References T1, T2, and Macromolecules, and will be given by Niklaus Zoos from the University of Zurich in Switzerland.
So good morning, everybody, and thanks for the invitation to talk here about quantification. And yeah, I'm really not a fast learner. So for me, it's always quite difficult to understand things based on presentations and learn things based on presentations. And the few things I think I understood is based on reading papers over and over and over again. So to make that possible to you too, I uploaded a list of all the papers on which my presentation is based on. So you can go to this link and you will find a list with all the papers I found interesting and useful for this presentation. And so the goal of the presentation is just to sharpen your awareness about things concerning quantification that might be useful for your projects as well. So first, I think it's important to repeat that quantification includes several steps, and the most important step is the acquisition itself. So here you can really avoid problems that later on are really difficult to solve or to get rid of. So here you have to think about a good water suppression and so on, a good choice of repetition time, echo time, and restriction of movement and all these things. And then in a second step, you have the pre-processing where we try to improve line shape and baseline. For the next step, the spectral analysis, where we try to, as Anke told us, to estimate the spectral peak areas. And this is important because the peak areas, they contain the information about the concentration. They are proportional to the number of spins included. And then finally, you come to the step which I will talk about here, where we correct the spectral area, peak areas for such things as relaxation, and we compare the signals to a reference signal. And this comparison is called data calibration. And this should lead to a good estimation of the amount of metabolite present in the investigated tissue. Ideally, we derive biochemical units such as molar or molar or molal concentrations. And you see it's a long way up there, so you have to do many things. But the idea that is you get a better insight when you do all these steps based on your data. And it's very comparable to a hike up to the Nielsen. That's a mountain where I grew up. So when you hike up there, and when you're up there, you have a really nice view to the Lake of Thun. So that's what I hope what you get when you do full quantification. There are also the macromolecules in this title, but I will not cover the macromolecules because I think this is more a part of the fitting. But I still have added some papers, some recent papers which I find interesting to the list I uploaded. So why do we need data calibration? Why do we need to compare our signals to a reference signal? This has to do with the signal detection in MRS. So, and this signal detection can be divided in roughly 3 steps. So in the first step, we create transversal magnetization And this step is highly dependent on B1 plus, the transmit field. Then we have T1, T2 relaxation, the magnetic field, even temperature can play a role, and then the localization sequence we choose, and so on. Many different parameters that play a role here. And then in a second step, this transverse magnetization induces an electromotive force into the receiving coil. And this step depends on the reception sensitivity of the coil at the given position. And then in the last step, this electromotive force is translated into a measurable voltage or current. And this step depends on the coil loading, the receiver gain, the cable, and even the position of the cable, and so on. So many more parameters that play a role here. And in order to be able to calculate concentrations based on the measured signals, we will need to know all these parameters, and we would not need to estimate the influence of these parameters onto our signals. And this is today, at least it's said it's not possible or not feasible. And so the easiest way to do that is to compare our signal with a reference signal. And here we have mainly two options. We can use either an internal reference that we measure from the same volume. So prominent examples are the water or the creatine signal, or we can use an external reference signal for comparison. Here we measure the reference signal in a phantom, where we know the concentration of the reference compound exactly. Now, in most publications, water is, I would say, in almost any publication, water or internal references are used for quantification. And why is that? So when you look at the acquisition of the metabolite signal and the reference signal, most of the factor can be approximately estimated to be the same. So the reception sensitivity and the coil loading and the B1 plus are the same if you acquire the reference signal from the same position. And that's why we can estimate the concentration based on these two ratios. So the ratio between the concentration and the signal is the same for our metabolite and also for the reference signal. And this allows us to determine the metabolite concentration using this equation here. And so if you have a good estimation of your reference metabolite, you can get a good estimation of your metabolite concentration. And that's the reason why creatine is not really recommended as an internal reference, because it's quite difficult to estimate the concentration of creatine in the given tissue or in the given voxel. But I still always look at the creatine ratio, because creatine has many advantages. It's not, in normal cases, not present in the CSF, so you don't need the CSF correction. And it's also, when you do something in pre-processing, the metabolites and creatine are treated almost equally in the pre-processing. So, and also when you, for example, you have an unsteady baseline in the fit, choline and creatine, they mostly affected both in the same way by this unsteady baseline. So you get a good feeling what is possible with your data. And so I still always look at the metabolite to creatine ratio. But to determine concentrations, the tissue water is recommended to use as internal reference. And with tissue water, you can use or you can calculate moles of metabolites per volume of brain tissue, so molar concentrations in a sense. Or you can calculate moles of metabolites per mass of brain water, molar concentration. I took these equations from the consensus paper, and I think it's important to know here that the molar concentration, which is used here, it's just the inverse of the molecular weight. So there is no assumption about the water densities in different tissue types when you use this equation. And especially if you measure your water signal at really short echo time and long repetition time, or if you have a good estimation of your fully relaxed water signal, this term is here, they vanish to 1. And you get an estimation of our molar concentrations without any assumptions about the water densities in different tissue types. That's different when you use molar concentration. So I think in most cases, the tissue water is really a good reference to determine concentrations. There are some potential issues. So for example, if we measure lesions in MS, for example, and we measure this at different time points, and we see changes from one time point to the next, we would still determine the correct biochemical concentration. So if we could cut out the lesion, but based on the MRS measurement alone, the interpretation is quite difficult. We wouldn't know if the metabolite has increased or the water has increased. And normally that's not an issue because water is really stable, but in these lesions, it might change. And then we have also a part of the water that is invisible. It's called MR invisible because it has a really short T2 time. And this part, this fraction of the water can also change in diseases. So that's why external references are used sometimes. So here you have the reference concentration, you know exactly, and based on this equation, you can calculate the concentration. But the problem here is that the acquisition that you do in vivo and in the phantom, they can differ significantly in certain parameters. So transmit field can be different, reception sensitivity can be different, coil loading can be different. So you need potentially a lot of corrections to correct for these differences in influencing factors. So you kind of gain certainty in the concentration, but you lose certainty in the acquisition. So a good way to correct for changes between the in vivo measurement and the phantom measurement, they are based on the principle of reciprocity. So for example, you can measure the current that is necessary to achieve a certain flip angle in the volume you want to measure. And at least at low fields, this is a direct measure of the sensitivity of the MRS experiment. I'm mainly working with Philips scanner, so there you can just do a proper voxel-based power optimization. And based on that, you get the drive scales or the voltage that is necessary to obtain a certain flip angle. And you can use this as a measure of the sensitivity for your experiment. And if you do that in vivo and in your phantom measurement, you can correct for the different differences in acquisition. So you get rid of the differences in coil loading with that and reception sensitivity. And because you did a proper voxel-based power optimization, you also can assume that the B1 plus is the same for both acquisition. So you end up just with the relaxation correction, which you can easily measure in the phantom at least. So that's an easy way to correct for these differences. But there are at 3Ts this assumption so that the B1 plus is equal to the B1 minus is not true. So you need corrections for that as well. So, but the question is anyhow, how far do we go? Like in the consensus paper, there is the recommendation to use molar or molar units always over institutional units. And it should be stated in any publication of findings what you are using. And I think it's important to realize that with MRS, we only achieve estimations of molar or molar concentrations. So even though we do comparison to a reference signal, we do correction for partial volume effect, relaxation effects, etc. We only get, we don't necessarily get concentrations that are easily comparable to other measurements, to other sequences, or to other scanners. So to illustrate that, I would like to show you some post-mortem data. I think 2 weeks ago, I measured them. So it's our measurements with long repetition time. It's about 10 seconds repetition time. It's an acquisition in the same subject, once with the vapor water suppression shown here, and in this here it's with metabolite cycling, so the water is not touched in this case. And overall, the spectra look quite similar, but when you look closely here, there is barely a peak at 2.2 ppm, and if you go down, there is quite a significant peak or a well-visible peak, and this peak is from acetone. And this is important for us to diagnose ketoacidosis in post-mortem cases. So, and this difference has nothing to do with compare, or you can not get rid of that by comparing it to a reference signal or relaxation, attenuation, correction, or things like that. So with that, I would like to shortly talk about the project that we have done at our institute, mainly to show you that with a good measurement, you can avoid many problems that you otherwise have to deal with later on. We measured 15 subjects post-mortem in CSF, and we tried to quantify ethanol concentrations. We derived gram per kilogram concentrations with water as internal reference, and we also used an external reference and did the corrections based on the principle of reciprocity. And we compared the measured concentrations with MRS, with biochemical measurements in the CSF taken out from the spinal canal, and we compared the concentration also with the blood ethanol concentration. And here is an overview of the results in black. So these are the cases where we actually found ethanol. So we didn't miss any case, that's good news. So in black is the blood alcohol concentration, and in yellow, it's the concentration determined in the CSF. And in between, we have the concentrations measured with MRS. And these two, they agree quite well. But compared to the CSF, we have deviations varying from 4 to 40%. And why is that? Just to show you here, this is a typical spectrum that we obtain from CSF. So we have mainly lactate here, and then the ethanol. And we've chosen metabolite cycling to measure in the CSF. We have chosen a repetition time of 3 seconds and an echo time of 30 milliseconds. And now that was probably not a good idea to choose this short repetition time. So when you look at this spectrum, it's also post-mortem data measured with a repetition time of 3 seconds, and then with a repetition time of 10 seconds. And you see the lactate signal here doesn't really change with the longer repetition time, but for the ethanol, you have a large increase. And when you look at the literature from rodents, then you see that the T1 of ethanol is quite long, so about 4,000 milliseconds. So in principle, you could correct for that by just adding a relaxation attenuation correction term. But then the problem is in post-mortem, especially in forensic settings, we have different temperatures, like varying from 5 to 30 degrees almost. And the temperature does influence the relaxation time, especially T1. And so to really be able to get the same concentrations as from the biochemical measurements, we would need to know the temperature dependence of our T1 relaxation of ethanol and then we could correct for each subject. But we could also have just increased the repetition time from the beginning. So, and that's my take-home message. So prevent problems already during acquisition. So that's very similar to what Michael has said yesterday about artifacts. So it's really good to think about your acquisition before you do it. And with that, I would like to thank the team at the Institute of Forensic Medicine, especially Dominique Gachot and Emma-Louise Kessler for her generous donation to our institute. And with that, I would like to thank you for your attention.
Do we have any question?
Yeah, thanks. That's a very interesting talk. You can't use the drive voltage to calibrate the system if you're using the body coil to transmit and you're using a head coil to receive.
You can. That's possible. Yes.
No, because the coil, which you don't know the loading is the same for the body coil and the head coil.
So you mean when you use the body coil to transmit?
Yeah.
And yeah, what we do is we measure the water with the body coil. So we measure the drive scale with the body coil, and we measure the water by transmit and receive the water signal with the body coil. And then we measure also the water with the reception with the receiving coil as well. So we do two different scans. So we do a transmit, receive with the body coil, receive, a transmit with the body coil, and receive with the reception coil. And then we do the actual measurements of the metabolite on the suppressed scan.
Yeah, okay, yeah, I need to think about that.
So, and then you can use the drive scale method as well. There is a paper around that describes it in detail. I think Peter Barker and his group did that.
Is that a quick one?
Yes.
Okay.
I've got a kind of hypothetical question. I'd like to know what do you think about using total metabolite signal as a reference to provide maybe kind of different information as a percentage of one metabolite versus the rest of the metabolite content as a sum of all the metabolites of the interest. Would it make any sense to use it as a reference because it would overcome some of the issues?
Can you say it again? Like you compare your metabolite signal to the whole spectrum, or what do you mean?
I would say that this would be a sum of all the metabolites from, let's say, LC model table.
Like the total sum of metabolites present.
Mm-hmm.
Okay. I never heard about that, to be honest. I think I'm just thinking about the CSF measurements. There, it would not work at all because, like, especially, I mean, it's a special case. It's post-mortem, but there you have changing concentrations in the CSF, like depending on the post-mortem interval, and so you would. And I don't think that, for example, there you would get meaningful results. And I don't know if in other cases you would. I don't know. I cannot say.
Okay, thank you for your comment. It needs some assumptions.
Nicolas, thank you for the presentation. I wanted to ask you two questions. So one is about the ex vivo measurements. When you measure on cadavers, and the brains were inside the body, or they were taken outside and put in a bag, and you measure it like that?
Yes, they are. Like, we are on the forensic side, so the goal is to obtain as much information out of the body without cutting it out.
So
Up.
there was no autopsy before the.
No autopsy before, so they come into the scanner like in one piece. In most cases, at least.
Okay, and the so that's the best spectral quality that you get. That's the typical best spectral quality in the spectra that you showed us. Like I was thinking that you could, I mean, spectral quality might be a bit better in terms of line width. Like, or
yes,
is it?
This is just I picked just one that was available. Yeah, it varies. Like. Yeah, that can change. Sometimes we have so we always measure the line width of the water, so it's between 7 to maybe 12 hertz, something like that.
And the second question, if I may, so you also have these nice papers with the electronic reference with the erratic. So what's your opinion after, let's say, a lot of experience that you have is among all of these three or ways of quantification, like internal reference, external reference with corrections based on correcting for B1, B minus, and then electronic reference. So you dropped completely this electronic reference. Is it and if so, why? Because it's not stable. There are some issues with hardware, or it's too complicated.
Yeah, it's so mainly it's about the hardware. You need to have the hardware, and at the Institute of Forensic Medicine, we don't have the hardware for erratic, and also like for me, or what I experience is that it's this reception sensitivity that's really an issue when you do use just erratic, because erratic mainly corrects for the differences in coil loading. And so you need something in addition to correct for the differences in reception sensitivity. So, and I did my PhD on erratic, and yeah, I spent quite a lot of time to get it stable. And there are sometimes somebody was running over the cable and then it wasn't stable anymore. So that's an issue that might be solved, but I prefer to use the drive scale method to, and I also have to say, like, even that, like I showed you, you need some corrections for these differences in acquisition. And I think the change that you expect in water in your study must be quite high to justify the additional effort that you have with external references. So I think that's also something you have to think about before you do your experiment.
Okay, very well. I would move on. Let's thank the speaker once more. Yeah. Okay, we'll be moving to the first selected abstract. It's entitled Investigating Glutamate T2 Relaxation Time, and it's given by Paulina Emilianova from the University of Manchester, UK.
Yeah. Well, thanks so much for the introduction. Well, once again, my name is Paulina. I'm a second year PhD student at the University of Manchester, UK. I would like to say, firstly, thank you so much to the organizers for the opportunity to show our work. And today I would like to present you our preliminary results on investigating glutamate T2 relaxation time. So glutamate is the major excitatory neurotransmitter in the brain with its natural abundance within approximately 10 to 12 millimolar range. Glutamate, for the most part, can be found in the cytosol of the cell, which is not actively influencing cellular activity in terms of firing, and that is mostly in neurons with a small contribution from astrocytes. Additionally, glutamate is found in vesicular compartment and to a much lesser extent in synaptic cleft and extracellular space. So currently there is a question mark over how much each of these compartments contribute to the overall MRS signal. And that is based on the evidence from in vitro studies indicating that glutamate bound to macromolecular structures within the vesicles may not be visible to proton spectroscopy because it has a very short T2 relaxation time. And here is an example of some of these studies. So it has been estimated that vesicular compartment could constitute up to 30% of total neurotransmitter in the brain. Additionally, recent modeling work by our research group proposing multi-compartmental biophysical model of neurotransmitter movement between glutamate compartments. By excluding contributions from the vesicular compartment, model simulations reliably predicted activity-dependent changes to functional MRS signal, which are consistent with reported empirical findings in the literature. And here is the paper by our research group on this model, if anyone is interested. And so far, I've presented you some in vitro and some in silico evidence that glutamate in vesicular compartment does not contribute to the MRS signal because it has a very short T2 relaxation time. There is no in vivo evidence to support this, and this is what our study was set out to do. So in order to measure a short glutamate compartment, it is necessary to use acquisition protocols with shorter echo times because they're more sensitive to detect compartments with ultra short T2 relaxation time. So far, most in vivo studies reporting glutamate T2 values are only sampling echo time within the range of 30 to 450 milliseconds. This is not adequate to detect compartments with ultra short T2 relaxation times. As such, our first aim was to examine whether there is a short T2 compartment of glutamate in vivo using multi-echo MRS measurements at short echo time. And depending on the outcome of aim 1, to determine T2 relaxation time for short and long echo compartments of glutamate. And lastly, to determine T2 relaxation curves for creatine and NAA. So this is how our study was set out using 3T Philips Achiever scanner and 17 healthy volunteers. We acquired T1 structural images for voxel placement. 8 milliliter voxel was positioned in the occipital cortex, centered along the midline and the calcarean sulcus. We acquired macromolecular data and metabolite data with water reference using single voxel STEAM. From now on, I will be referring to macromolecular data as MM and to metabolite data as MRS. We acquired MM data for 10 participants over 5 echo times of 9.3, 13, 20, 35, and 70 milliseconds. We've used inversion time of 600 milliseconds, repetition time of 2000 milliseconds, and 16 averages per echo time. MRS data was acquired over 7 echo times as indicated in the diagram. Can I show you? Yeah, so, and we use the following parameters. Repetition time was the same, 2000 milliseconds. Mixed time was 16 milliseconds and 4 averages per echo time. So for the remaining 7 participants, we only acquired MRS data over 7 echo times. Just a note, echo time was randomized for all participants. Another key component of the study was to investigate whether the T2 curve changes with activation compared to rest. However, this data is still being analyzed and is not shown today. So that you can see how our study was set up, this is the paradigm we used. This is directly taken from a paper by Bettina Eep, and here is a reference if you would like to look it up. So our during MRS data acquisition, our participants were visually stimulated. Our experimental design consisted of 7 conditions, 1 condition per echo time. 1 condition had 8 trials of interleaved rest and visual stimulation, and 1 trial lasted 64 seconds, meaning that we had 256 seconds of rest and 256 seconds of visual stimulation. So during rest period, participants viewed black screen with white fixation dot in the middle, and during a visual stimulation, participants viewed flashing checkerboards and completed a task to maintain attention levels. Just as a reminder, today we're only presenting new results for our rest data, and visual stimulation data is not shown. So MM data were analyzed in GMR UI And our pipeline included frequency alignment, signal averaging, HLCT water removal, and referencing to NAA. Reduced T-dependent basis sets were simulated in an MR scope and included 4 larger signals of creatine, NAA, glutamate, and glyphosphorylcholine to account for choline-containing compounds. And this figure here is showing an example of glutamate, creatine, and NAA simulated over 5 echotimes. So residual cleaning and fitting was done in quasar Y, where simulated signals were subtracted from the spectrum. Then we used HLSVD to parameterize spectrum, excluding components outside the fitting range and adding up the remaining components to get a participant-specific noise-free spectrum for each participant. So after visual quality inspection, we averaged the remaining spectra across participants for each echotime, which gave us a noise-free group mean spectrum. And here's an example of noise-free group mean spectrum across 5 echotimes. Similarly, MRS data was analyzed in GMR-UI. It also went through quality assessment involving signal-to-noise ratio, NA line-based, serial-based, and visual inspection. If data were 2 standard deviations away from the mean or serial base were greater than 25%, data were excluded from the further analysis. T-dependent basis sets for this analysis included 6 signals of creatine, NA, GPC, glutamate, glutamine, and minoxidil. And here is an example of creatine, glutamate, and NA simulated over 8 echotimes. MRS fitting and quantification was done in quasar Y. The contributions of macromolecules was addressed by including in vivo signals in the T-dependent basis sets below 100 milliseconds. This is an example of in vivo spectrum depicted in red for a single participant with model feed depicted in blue. And this is a large example of a spectrum collected over echotime 9.3 milliseconds. So the T2 curve was fitted in MATLAB. Prior fitting data was normalized for each participant to the first data point and averaged across participants for each echotime. And data then was T1 corrected and fitted using fit and alarm function in MATLAB. Mono-exponential, bi-exponential feeds were assessed using IKK criterion for glutamate, creatine, and NAA, and the results were slightly surprising. We found that the data fitted significantly better with more complex model for all 3 metabolites, as indicated by IKK values highlighted in blue. So feed parameters indicate the existence of 2 compartments. What we found was for glutamate, the T2 for short compartment was 22 milliseconds, and T2 for long compartment was 197 milliseconds. For creatine, we found the T2 for short compartment was 31 milliseconds and 137 milliseconds for the long one. And for NAA, we found the T2 for short compartment was 14 milliseconds and 186 for the long one. So while we are excited to see by exponential decay for glutamate with components corresponding to cytosolic and vesicular compartments, we did not expect to find by exponential decay for creatine and NAA. So although surprising, we found some preliminary evidence in the literature that NAA collected at short echo was also showing by exponential results. And it was suggested that NAA collected in vivo could arise from neuronal and myelinic compartments. And short T2 compartment was attributed to NAA located in oligodendrocytes. So in terms of creatine, I guess one of the potential explanations could be that we did not fully account for contributions. We can probably see some And just as a reminder, earlier in this talk, it was mentioned that most of the studies reporting T2 values for glutamate sampled echotime within the range of 30 to 450 milliseconds, which is not adequate to see compartments with short T2 relaxation times. In our experimental study, our minimum echotime was 9.3 milliseconds, which should be more sensitive to detect compartments with short T2 relaxation times. And to summarize, these results provide preliminary evidence of by exponential decay for glutamate, creatine, and NAA in visual cortex at 3T, However, for NAA and creatine, there should be explanation other than vesicular compartment. So our findings for the long T2 compartments for 3 metabolites are in agreement with empirical literature using mono-exponential feed and a minimum T of 30 milliseconds. And our future work will incorporate new analysis methods, such as, for example, fitting of each resonance component independently, excuse me, to examine resonance-specific T2 relaxation time for 3 metabolites. And I would like to say thank you so much to Dr. Caroline Lee-Cornell, Professor Steve Williams, Dr. Laura Parks, and the rest of the team for the invaluable help with this project. And well, thank you for your attention.
Go ahead.
Well, you asked me yesterday about some of this.
Yes.
Just one comment, the one reference you referred to was the NAA measured exponentially by Erin.
Yes.
In Vancouver. That actually, I looked it up last night. We redid those data analysis and doing a simultaneous fit of all the data. I'm afraid the second compartment was not large enough to persist. So that's why it was never published. It was only an abstract.
Yeah, okay, thank you so much.
But anyway, I mean, I think it's very interesting. Obviously, you're right to suspect many other effects that could induce bi-exponential. I mean, you easily get bi-exponential with artifacts, with not properly getting rid of the residues in your macromolecules and stuff like that. But on the other hand, Kadir, who's sitting here, looked at macromolecular baseline defined by diffusion and compared with macromolecules defined by inversion recovery. And we found some differences, also very preliminary data, but that almost looked like there were some metabolite resonances that were broader, and that would sort of correspond to shorter T2. So it could be real, but I would not bet anything on it at this point.
Yes, thank you so much. I understand. Thank you.
Hello, this was super interesting. Thank you.
Oh, thank you.
So I'm interested in glutamate and like what we are measuring. But like in Bettina's paper, I know that glutamate was like really high in the beginning of the blocks. Did you account for that because of like the flickering checkerboard?
Well, we've not finished analysis yet, so we're only looking at the rest data. So I'll probably will need to come back to you on that.
Okay, sure.
Yeah, I'm more than happy to stay in touch by email if you'd like.
Okay, cool. Yeah, because I think like that may influence the results a little bit. If you also find high glutamate in the beginning, then no.
And I would like to introduce the first speaker. This is Bernhard Lanz here from the CIBM, the EPFL here in Lausanne. And I've been knowing Bernhard for many years. To see his growth as a scientist has been amazing. And he will talk about preclinical MRS today to us. Bernhard, the floor is yours.
Thank you very much. So it will be hard for me to keep with the Swiss reputation of being on time, since I should manage to make my presentation in -5 minutes. I would like to thank the organizer for giving me the opportunity to present today. And I should present on preclinical MRS, which is, of course, a very broad topic. So first of all, I would like to remember what is actually preclinical MRS. So it could be defined as MRS applied to animal models for scientific questions that cannot be probed directly with human MRS. And the scope of this presentation will focus on rodents, so rats and mice, and on applications to brain studies. So the essential part of what I will present is taken from the consensus paper we organized on preclinical MRS on the rodent brain. So from the initiative of the editors Nyong Choi and Roland Kreis. So I would like also to take the opportunity to thank all the co-authors who contributed to this consensus paper and the supporting group as well. Many of them are present here, and it's a great pleasure for us to have you here. So why do we do preclinical MRS? So actually, there are different usefulness for preclinical MRS in rats and mice in most cases. So first we have to think about the ethical considerations, which is more and more of a topic, because ultimately the goal we have is not to find ways to cure rodents. So there is one usefulness in terms of biological aspects with animal models. That's one aspect I will talk about. And the second aspect is actually the usefulness in terms of MR physical aspects. So the new MR acquisition approaches we can develop in preclinical MRS. So why do we do preclinical MRS in terms of biological aspects? So rats and mice are actually good biological models of tissue development, function, and metabolism for the human. And they are mimicking multiple aspects of human disease, especially now with all the transgenic mice models available. And actually, there is something quite fascinating especially for physicists, and it was fascinating me also at the time. I chose to go the way of biophysics, what we call biophysics, although life has not really a definition in physics. So living tissue works as a whole and not as the sum of its components. And that's the reason why, actually, as discussed also yesterday during the presentation by Steve Williams, cell cultures cannot be used. They can be used for basic cellular metabolic questions, but actually in vivo studies are key for more complex organ functions. And I like to say from a more engineering point of view that we can't really apply a kind of reverse engineering like you would do, for example, to understand how a plane works. You could basically decompose the problem, learn how the engine works, how the wings work, how the body works, and then the different parts of the plane, and then put everything together, and you would understand how the plane flies. And that's not really possible with in vivo organs, especially the brain. And then MRS is a non-invasive technique, so it has also the advantage that we can characterize disease progression and effect of treatments with longitudinal studies. In terms of physical aspects, we have actually the availability of higher magnetic field strength in preclinical studies, and that enables to study with higher sensitivity and spectral resolution. And we typically use also anesthetics, and we come back to that, which enables longer experimental times and with minimal motion artifacts and instabilities. And with that, we can push some studies much further in terms of duration, like carbon-13 MRS, for example, where we can go deeper into the biochemical pathways we analyze. We have also lower restrictions technically in terms of gradients and RF limits, and that enable the developments of new MR approaches at lower risk. In terms of physiology and anesthesia, so we have actually we have to consider different aspects for the experimental design. So the first one I was talking just before is the anesthesia, which is critical for in vivo experiments. So it decreases the stress of the animals, the potential pain in case of surgical intervention, and the biological motions. But on the other hand, it has also other effects, side effects, like it decreases the respiration rate, the cardiac activity, and those things have to be taken into account, among others. There are very few studies actually of MRS in awake rodents. We report as well in the consensus papers. They are very challenging. They require long training periods for the animals. And then they should be also done with care, monitoring the cortisol levels, for example, to be sure that the animal is not under extreme stress. So in the consensus paper, we have a table summarizing the major anesthetics used in preclinical MRS and also the potential effects they might have on the metabolites you would like to measure. And that's something to take into account prior to any experimental design. There are other pre-anesthetic considerations like stress, strain, sex, cardiac cycles, weight, and age of the animals, which will change the effectiveness of the anesthesia and have a direct impact on the MRS measurements and the neurochemical profile. So the correct dosage of the anesthetic will depend on that, and it should also provide good sedation and analgesia. And then the monitoring and the recording of at least of the respiration rate and the temperature of the animals, but ideally of more physiological parameters, is key to ensure that the measurements are done in good conditions which represent normal physiology. So on the more pragmatic side, in terms of subject feedback, what is the practical difference for the experimentalist to having worked both on human MRS and preclinical MRS? We have a quite substantial difference on what we can get back from the subject during the scan. So on human MRI scans, we typically rarely perform longer scans than 45 minutes in total, typically split in 10-minute scan blocks. And you have a direct feedback from the subject. So basically you have an intercom and you can ask the subject how it's going, and you can also motivate them to stay still. And you have basically a feedback loop. When actually on rodent studies, well, luckily the manufacturers are not providing an intercom, because I would be very worried if I would get a reply. But that's something we have to take into account. So we scan over extended periods, but we don't have a direct feedback of the subject. So the animal is invisible in the center of the magnet. That's a limiting factor. So I would say it's the preclinical MRS equivalent of the Schrödinger's cat experiments, which was a very hot topic at the beginning of the quantum mechanics interpretation. And you know, with the problem of the cat being either dead or alive or in a superposition state. And in our case, we really would like to have our animal alive, definitely not dead, and we would not know what to do with the superposition state. So it's quite funny as well when you go into the history of this, because originally, so Schrödinger must have something against cats, because when he was talking with Einstein about it, Einstein was mentioning about a barrel of gunpowder that to show basically the paradox. So it should be either exploded or not. And when he gave his idea to Schrödinger back, it was like, oh yes, let's just put the cat on the side of the barrel. So a key thing now is, as I mentioned, is that basically we need to get some physiological feedback from the animal. So typically we recommend at least to measure the body temperature, which drops significantly under anesthesia, and the respiration, but the more parameters you can get, the better. In terms of hardware, the main differences we have with the human MRS is, of course, the small size of the brain of a rat and mouse. So we see it here in comparison on the picture. So it's extremely small. So it has a very strong B0 inhomogeneity and a lot of air tissue interfaces in a small volume. And so for that reason, the small volume of interest we're interested in require ultra high field systems. And they also will need higher gradient strength. So typically we work with gradient strength stronger than 400 millitesla per meter. And we also need very good shimming methods to obtain a reasonable line width in our measurements. On the RF side, so we have actually lower RF power requirements than in humans because everything is smaller and the coils are more efficient. We typically work with the surface coils, which provide a higher SNR for a region close to the coil, but they are, of course, especially inhomogeneous. We can partially compensate that with adiabatic RF pulses. And contrarily to humans, we have actually legally unlimited B1 and strong gradients, gradient strength available. So we could choose theoretically optimally what we want to do in our MRS sequence. However, although it's legally possible, we should still remember that the problem is still there. We still have tissue heating, potentially tissue heating issues, especially at ultra high field, and that's something you should always be careful with, and some things that we observed, for example, at 14 Tesla. Also on the RF aspects, there is something specific to preclinical studies. So in this case, with the small sample volumes we have, the noise in the measurement is dominated by the noise of the coil and the receive pathway. So cryogenically cooled RF coils are a good option to increase substantially the signal to noise ratio. So we have an example here we show also in the consensus paper, which shows a 5.2 increase in SNR. So in terms of proton MRS, we typically go for ultra short echo times, shorter than 10 milliseconds. They are possible to achieve with the very high B1s that we can have with our RF coils. They are used preferentially so that we get more accurate quantitative information with minimal J evolution and T2 losses. And we use relatively long TR to avoid T1 effects. Then a key factor is actually the localization performance of your sequence. So of course it should be able to detect signal originating from the volume of interest, but at the same time also to suppress signal from the outside of the volume of interest. It should have minimal chemical shift displacement error related to the bandwidth of the localization pulses. It should be insensitive to B1 inhomogeneity, ideally, especially when using surface coils, and efficient for water suppression and to eliminate, of course, the strong water signal. So what we recommend in the consensus are actually those three sequences: STEAM, spatial, and laser, with examples here shown in the rodent brain. We also summarize the performances of those three sequences in a table, although it should be mentioned that the performance here are based on in-house optimized sequences and not the vendor's versions you get out of the box, which typically need a lot of the optimization before they perform best. So this is an example of a proton spectrum you can obtain at 9.4 Tesla. And we get more than 18 markers of myelination, energy metabolism, osmoregulation, neurotransmitter metabolism, antioxidants. And that's actually a very good, as I mentioned, something, an extremely valuable marker, especially for longitudinal studies on mouse model of disease. Now, a question that can arise is actually about MRSI. So there is not much about MRSI in preclinical models. And the main reasons behind it is actually the difficulties to do it. So because we need to shim over a large volume, and we have many tissue interfaces in a small volume, the limited SNR because of the small volume, and also the partial volume of muscle, skin, and fat, which is substantially higher in the head of mice and rats. So basically the proportion of the volume taken by the brain in the head is much, much smaller in a rodent than in humans. So it's challenging to implement in terms of shim, water suppression, lipid contamination, also particularly for long measurement times and for absolute quantification. However, as we already saw yesterday, research is ongoing on the topic, and there are substantial progresses done recently to make proton MRSI move on in preclinical studies. So we also touch on the phosphorus spectroscopy in the consensus paper, which is a complementary technique to proton MRS and carbon-13 MRS. It can probe energy metabolism, creatine kinase and ATP synthase in a dynamic experiment with magnetization and saturation transfer, can measure the pH and the magnesium concentration as well. It has a lot of challenges as well because it has a low gyromagnetic ratio, 2.5 times lower than protons, so it requires ultra-high field systems typically. It has short T2 as well, so the components have short T2, which basically rules out echo-based approaches like steam, press, or laser. And then there is a large frequency range of the phosphorus resonances, so we need broadband pulses. The two main methods that we recommend in the consensus paper are 3D ISIS, which is basically a zero echo time sequence, but it's sensitive to motion. It can be made fully adiabatic as well for surface coils. And OVS-based localization, or actually even a combination of the two. And you see some examples on the side how actually we get localized signal, which is not affected anymore by the signal you get from the skull and the muscle. And if you manage to distinguish clearly the inorganic phosphate resonance at 4.9 ppm and 5.3, that's a good signature of excellent spectral quality, both in terms of shim and SNR. Then there is carbon-13 MRS, which was also widely applied in preclinical as compared to human MRS. So the main reason behind it is actually that most of the, well, a lot of energy metabolism is based on exchanges of carbon chains. And also the natural abundance of carbon-13 is very low, is 1.1%. So it can be used as a tracer for energy metabolism. So typically we use glucose, which is the main energy substrate for the brain. So we have used, in the past, people have used 1,6 labeled glucose or the cheaper C1 labeled glucose to measure neuroglial metabolism and the apparent neurotransmission between glutamate and glutamine. Labeled acetate can be used as well to be more specific for glial metabolism, or C2 glucose for pyruvate carboxylase measurements. So similar to phosphorus, there are some limitations, physical limitations based on the nucleus. So it has a low gyromagnetic ratio, about 4 times lower than the proton. So we need ultra-high field systems. It has also a large frequency range for the metabolites, about 30 ppm, which is about 3000 hertz at 9.4 Tesla. So it's very nice on the receive side because we have very distinct peaks to quantify, but actually on the transmit side, it's a bit more of a problem because we need broadband pulses. So the two main approaches we recommend for carbon-13 MRS in preclinical is actually our indirect POCE approaches. So where you measure the presence of a carbon-13 on the proton resonance with carbon editing, or polarization transfer approaches. So we have two examples here, like the bicep spectral sequence and the IZ-STEP for indirect and direct detection. So indirect detection is typically used for smaller volumes due to the increased sensitivity of the proton measurement, and polarization transfer methods are used for the extending biochemical information you can get from the direct carbon spectra. So as we see here, for example, so this was a measurement done at 14 Tesla here in Lausanne on a mouse brain, and you can see how rich information we can get, actually a lot of different carbon-13 positions, glutamate, glutamine, and other brain metabolites. And this was measured with a homemade surface coil proton carbon. When you get all those measurements, at the end, you also want to do it dynamically. So the goal is to see how the spectra are growing over time, and applying a kind of two-compartment model approach where you basically summarize the energy metabolism of the brain and classify cells into subcategories, like here in glial cells and neuronal cells. You have three parameters, which are the biochemical fluxes, and you can actually quantify them directly in terms of micromole per gram per minute by adjusting the simulated result of the model to the turnover curves you get on the glutamate, glutamine, aspartate, and other metabolite carbon position. I also wanted to briefly talk about hyperpolarized carbon-13. So it's not something which was covered in the consensus paper because we thought it would definitely need a separate paper for it. And but tomorrow we will have a session on X nuclei, which involves a lot of hyperpolarization and hyperpolarized carbon-13 as well. So as you will see, it generates a very strong enhancement of the signal you measure, but it has a short lifetime in terms of T1 relaxation. So when you try to model it and get information in a similar way you would do with thermally polarized carbon-13, you get actually rate constants, which are a signature of many aspects. So you have actually the metabolic isotope exchanges themselves, but then you have magnetization decay through the successive pulsing and the T1 relaxation of the compounds. So, and ultimately the target we would like to have is the measurement of the metabolic isotope exchanges in terms of fluxes in micromole per gram per minute. And that's still something that needs to be, I mean, we need to work on to get quantitative information out of those experiments. So finally, in the consensus paper, we give a series of recommendations. So if you're intending to start preclinical MRS, it's advisable to have a look through those suggestions to avoid going into, well, trying, let's say, less successful approaches. And with that, I would like to thank you for your attention. And I would like also to thank again the co-authors of the consensus paper and the supporting group and all my colleagues from Lausanne working on preclinical MRS. So thank you very much.
Yeah, Bernard, thank you very much for this great overview. The talk is open for questions.
Very nice presentation. I have more of a comment than a question.
Yeah.
I think, you know, like I talked to several people in the past, including, and Noam Shemesh made a comment that the term preclinical potentially does a disservice to the field. I think it's a very important field with beautiful work. If you call it preclinical, it almost sounds like you got to justify why you're doing what you're doing. I mean, I saw like several slides where it said why, you know, and you described it beautifully, but I feel like it's almost like, you know, should have a stronger chest and kind of say, look, we do this and we can do beautiful things with it. We learn about biology. Yes, preclinical is part of it, but it's only a part. And I really, you know, admire this work.
Yeah, thank you very much. It's actually a very strong point, and we have to justify the reason why I went on this question is that every time we need ethical authorization to do an experiment, we get all those kind of questions. Why do we do it on animals? And sometimes it's almost funny because, I mean, the experiments we do are quite challenging in terms of maintaining the physiology over longer periods. And And when you get the questions, why don't we do it on humans? I mean, I don't think everyone would like to stay 8 hours in a magnet and do those kind of experiments. So of course, it's a trade-off. And I think ethically nowadays, it's more and more important. We also had cases in Switzerland very recently where we had to vote for, there was an initiative, a popular initiative to just forbid any animal experiments. So it's something that we need, I think, to talk about more on our side to really show the people what we can do with it and why we need it, why we can't directly go for human MRS, for example. Thank you.
Are there SAR regulations for doing animal studies? Because SAR regulations.
Yep.
So, because I did animal studies 30 years ago, and up till today, I regret that I never did any SAR computation. Is this done?
No, that's true. Actually, we don't do it, and there are no legal requirements for it, so people tend not to do it. But we, as I mentioned, we have experiments with quite, say, heavy cases in terms of energy depositions where you see effects of SAR problems, and something we should consider in the future just for the well-being of the animal and also for the success of the experiment, because at the end, we don't want to cook the animal.
Because sometimes I get doctors, they read an animal study and they say, well, this is brilliant. But then I say, yeah, it's a high field, and most probably you can do with animals stuff you cannot do with humans.
Right, yeah.
But would there also be let's say 5 watt per kilo? Do you think that there will be other limits?
I don't know. Well, I think it should require a proper study, basically, to find out where the limit is. Of course, the surface to volume ratio in those animals is very different. So we could expect that the thermal exchange is better. So probably we could have a bit more of energy deposition, but it's something that needs tests, I think. Yeah.
Thank you.
Thank you very much.
It's actually just a comment picking up on why we call it preclinical. Yeah. One reason is so we can talk about it in the pub. Because if you talk about it in the pub and say, oh, remember that rat we did yesterday, and someone overhears you, then you can be in trouble. Whereas you say, you know, we use this preclinical sequence, then, I mean, so, I mean, I take the point of the previous comment that, you know, we shouldn't be, if we're embarrassed about doing animal experiments, we shouldn't be doing them, you know, but at the same time, you don't want to lay yourself open to people with fixed ideas,
yes,
deciding to take it out on you.
Yes, thank you for this comment. Yeah, and actually it's a very strong point. And personally, I had very big trouble starting in the field, to be honest, and to convince myself that we are doing more good things at the end where we get really strong results out of it, which are worth the sacrifice, basically. And it's true that the advertisement people make against animal experimentation is pretty tough. So I received some flyers also, like before this initiative as well I was mentioning about, which describe us as devils, basically just working for their own career and ready to kill thousands of animals just for their private interest. So it's quite it's something we need more communication, but I'm not quite sure who needs to communicate more at which level. Because for us researchers, I mean, we are, well, it's maybe a bit difficult to have all the connections to probably the institutions themselves should do this kind of work with the help of the researchers.
Sounds great. Actually, I have a quick question, Bernard, because I know you have been doing also clinical MRS, or let's say in human studies, and I guess preclinical, if you want to still call it like this, and clinical MRS should benefit from each other. Have you been involved in a study that set out, let's say, these two separate paths, but kind of in a simultaneous organization and planning of a study? So that we say we do a human study on this part and then a preclinical part to investigate another aspect? Especially in carbon-13 MRS, in which I was involved the most, basically the knowledge we can gain on the rodents is a lot. I mean, it's a big part of the game, let's say, and so we can perform much longer experiments. We can get much more information on the deeper biochemical network, and then we can use those kind of knowledge to design the models we can apply in humans for shorter acquisitions, because of course you have less information, less dynamic information, so you need to constrain somehow your models.
and the knowledge we can gain on rodents just on the biochemistry itself without thinking about disease yet helps a lot in that sense. Yeah.
Right.
And actually, just to say, we had actually to remove the word preclinical from the title of the consensus to be more specific and really mention about rodent brain, because it was mentioned by the reviewers that it's too vague. So I think, yeah, preclinical is maybe a bit misleading. Yeah.
Well, thank you very much again. Let's thank the speaker again. And we will move on to the second talk of this session. And we are very happy to welcome Itamar Ronan, who recently moved to the University of Sussex in the UK. And I think we can call him the father of diffusion-weighted MRS, or at least one of the main driving forces behind them. And today his talk is entitled MRS, Translational Work, Bench-bedside, or which comes first? I guess the cow or the egg. And we look forward to your presentation. Thank you, Itamar.
Thank you very much, Ralph and Alireza, for moderating the session. Beautiful to see you again. Beautiful to see everyone again, actually, in this beautiful hall after a hiatus of several years. Christina, thanks so much for leading us the way and whipping us into order into this magnificent workshop. So really wonderful to be here. In Hebrew, we say, by the way, that success has many fathers, whether the failure is an orphan. So yeah, I'm glad to be one of the many, many fathers. And actually, I think in the order of events, I'm actually quite a young father in diffusion-weighted spectroscopy. The field started way before I looked even at science at all. But I'm glad to be part of this sequence of contributing to that off the beaten track research field, which, by the way, was not supposed to be my topic today because Christina told me, sorry, we don't have time for diffusion-weighted MRS. But you're talking to me, so diffusion-weighted MRS will find its way into the talk sooner or later. But for now, we are going to do a kind of ruminatory type of examination of the link between preclinical, wait a moment, I'm not going to use that word. By the way, I want to remind everyone that the word nuclear was taken away from us at some point, sooner or later, exactly because I think of the pub conversation type of argumentation. So we are in magnetic resonance spectroscopy, and we're talking today about the interactions between the various modalities or the various models that eventually bring the work to its application in humans. So yeah, what comes first? Oh, sorry, there was a mixed metaphor here. Here is the correct metaphor. Now the chicken is back in order. A little bit of anatomy of today's talk. We'll talk about, since in the title there was bench and bedside and translational, I would like to a little bit clarify how do those terms relate to the work that we are engaged in. And we will talk about it in the context of the cross model. That's a new term that I took the liberty to introduce. Cross modal, not model, but model MRS. And I'll just go through a few examples, some of other people's work, and then the diffusion-weighted MRS. The second shoe will fall with the diffusion-weighted MRS eventually. And since the word translation is in the title, I'll try to engage you in trying to think about how can we actually work better to achieve better translation of our methods. And here, if I may, I would like to make a small suggestion that at the end of my talk, rather than asking me the questions, maybe we can have kind of a more of a forum type questions. So if people have questions, they can ask them. And then if I can answer, I'll be glad to. But if there are other people that are here, and many of the experts of the works that I'll be showing here are in the audience, are more than welcome to just go to the microphone and give their answers. So translational work in MR in general, or MRS, or in empirical science, I should say, in theory and in practice. So everything starts from an impetus, kind of an urge to bring something to the empirical world. And it can be a discovery, either from MR world or from outside the MR world. And since we're talking about specifically about things that are related to clinical problems or pathologies, they are typically related to some sort of a pathomechanism that will eventually lead to a detectable change in a MR spectrum. It can be proton, it can be phosphorus or C13 or DMI, for example, or any of the modalities that we deal with. And then if we think about this linear type of process, if we buy into this scheme, then we have the bench, and in the bench we have a choice of appropriate model system that would represent the disease that we want to look at, and we will either adopt or develop old or new methods to detect this change that matches with the pathology, and we will try to substantiate it, of course, with non-MR measurements, with histology, immunohistochemistry, and so on and so forth. And that ideally would lead eventually to the bedside. And the bedside, we will choose an appropriate protocol for the human scan. We'll do a proof of concept scan, typically patient versus controls, and then we'll develop a more kind of advanced experiment. We'll talk about certification of different phenotypes of the pathology, or look at disease progression or response to therapy, or we'll add other non-MR correlates or MR correlates that are not spectroscopy, all in the hope that we'll eventually will reach the golden goal and implement in clinical care. But we know that all this is, well, for the lack of a better term, BS, because we know that everything is interlinked between each other. And we cannot really have this kind of linear timeline that goes from idea to bench to clinical applications. And the order of things are really sometimes serendipitous and happen in a very miraculous way. And sometimes it weighs towards a stronger side of this triangle, and sometimes it is equilibrated among all of those things, and sometimes goes in a circle whereby the bedside provides more ideas to the impetus, and so on and so forth. So this is, I think, one of the strongest take-home messages that I would like us to go home with. And I will start with the kind of, I should say, the kind of bread and butter of clinical MRS, which is MRS in cancer, which is probably the most widespread application, clinical application of MRS, which is to look at tumors in various reasons, for discriminate tumors, from tissue, and of course to grade tumors according to their severity. We know that there are certain parameters in the standard proton spectra that would lead us to hypothesize about links with pathological mechanisms that are behind the tumor progress. And there is this beautiful paper from Berlin and co-authors that kind of summarizes all the clinical uses of MRS. And she actually put in the paper, the beautiful paper from Franklin Howe, that actually takes this kind of very nice statistical overlay of spectra of various tumors, and to show the difference from just standard proton spectrum from parietal white matter. And let's take a look a little bit about the timeline of how things developed with respect to MRS in cancer. So we can start with Steve Williams with his work in tissue extracts. And here there are tissue extracts from samples from human tumors. And we can see the extracts showing normal white matter and grade 3 astrocytoma, added with a marker to calibrate the spectrum with respect to its ppm values. And this would be, let's say, one of those bench works that we're looking at, substantiate very properly in a kind of a tissue extract way, what do we actually have in the tumor? Then we have the work from Decaux in our friends in Grenoble, in which there is an animal model. So we moved from bench to animal models. And here there is a model of intracerebral glioma in the rat, and there are time points in which this in vivo spectrum were taken, and the regular evolution of the increase in choline and decrease in NAA and increase in lactate and lipids that you can see across several weeks in the tumor. And then finally, we have the human work from Jens Pham and the group that looked in vivo, in humans, in astrocytoma and oligodendroglioma. And you see those beautiful results. And all we need to do now is look at the timeline. But the timeline is really surprising, because this is from 1989, and this is from 1989, and this is from 1989. So it's all the same year. So how does that happen? There is no timeline bench to anything. So everything happened at once. And the conclusion is that 1989 was a very good vintage year for MRS in cancer. And yesterday I actually looked, and it's also for red Bordeaux, Sauternes, Burgundy-Rhône, and possibly some Alsace, definitely Barolo, and absolutely the sweet Riesling of the Moselle. So there is lots of reason to look at 1989 as a really very good year. And the take-home message from here is that really there is no clear bench-to-bedside timeline in MRS discovery and research. And one of the reasons is that this is a non-invasive tool. So we can, if a researcher wakes up in the morning and says, I really want to look at this particular pathology, all they need to do is to have a good hypothesis, a good method, and bring them all together and recruit the patients. In parallel, there would be another group that does the preclinical work, the animal work, or the tissue model work. And in principle, they would all support each other to eventually bring us to know better about the link between what we see in the spectrum and what we have in the pathology. So ideally, we are looking for strong interaction among the bench and bedside, and it is crucial, I think, for a successful implementation and dissemination of methodology across platform. Now, I will move to the second example, which is a more specific example. And again, some of the heroes of this story are here in the audience. And the story is the usability of 2-hydroxyglutarate in MRS to look at brain tumors. And it all started with a humble paper in Acta Neurophysiologica that talks about the analysis of the IDH1 codon 132 mutation in brain tumors. Now, this terminology, until 3 days ago, I knew nothing about. So panicking a little bit, kind of looked more into it. And of course, one look and finds quite quickly that IDH1 and 2 are isocitrate dehydrogenase genes that express IDH or isocitrate dehydrogenase. And the question is, what is isocitrate dehydrogenase? And this is an enzyme that catalyzes the oxidative decarboxylation of isocitrate to alpha-ketoglutarate. And it seems that in many tumors, IDH1 and IDH2 are overexpressed or expressed more than in healthy tissue, but that the expression can be in a wild type or in a mutated form of the gene. And it seems that the very high frequency of IDH1 mutations in grade 2 astrocytic and oligodendroglial glioma suggests a role in early tumor development. And this is just a cut and paste from the abstract of this paper. A year later, in 2009, already came a kind of a more comprehensive link to clinical outcome of this finding. And this is the New England Journal of Medicine paper, IDH1 and IDH2 mutations in gliomas. And here again, quoting from the abstract, the tumors with IDH1 or IDH2 mutations had distinctive genetic and clinical characteristics. Patients with such tumors had a better outcome than those with wild type IDH genes. And this figure actually tells the entire story of really by how much the survival rate of the patients with the mutated IDH format had a better survival than the wild type gene. And this is in the case of glioblastoma and in the case of anaplastic astrocytoma. But the bomb fell just a little bit later, is that the IDH1 mutations produce a small molecule or a metabolite that is called 2-hydroxyglutarate. That is something that makes MRS people stop sleeping at night and thinking, what can we do with it? And this is, again, from this particular Nature paper from Lenny Dang and co-authors. And this is just to show you about this little molecule that is now the target of people that do in vivo MRS. And if we are constructing the timeline, the fake timeline from animal research to humans, we should mention a paper that did preclinical work in 2012. And this is the paper from the group in UCLA. And this is, again, we are not here to pass judgment, but we are here to tell a history. But sometimes we also look back at history and learn a little bit from mistakes in history or things that could have been done better. And this is just, for example, a spectrum that is taken from a knockout mouse with a 3mm optic glioma. And this mouse was injected with a 0.1 cc of 1 molar of 2-hydroxyglutarate to show that there is a difference in the spectrum that is acquired after the tumor has uptaken 2-HG and not generated endogenously. And what can I say? I mean, we are now in 2022 and we're looking at spectra, and there are consensus spectra papers about quality of spectra. And without, again, passing too much judgment, this kind of spectrum obviously does not pass the quality that we typically require from spectra to be kind of our guidelines into making decisions about quantification. And then there is quantification. And again, if you look a little bit more carefully at the numbers, there is something that doesn't really compute in terms of the concentrations of 2-HG versus glutamate. And everything looks a little bit as if it requires a little bit of a more tender care and love in terms of how the experiment should be done. And this was done in parallel, of course, by the man that is sitting right there no, it's not. So Ovidio and his colleagues in MGH actually bypassed the preclinical question and went directly into a very, very meticulous setup of experimental method that will first of all substantiate the detectability of 2-HG with spectral editing, and then applying the same method to actually detect it in tumors while taking as control groups tissue that is not affected by tumor or tumors that do not contain the mutated IDH gene. So first of all, 2D COSY experiments that identify the cross peak that is related to 2-HG, editing that indeed shows that at 4 ppm, or roughly at 4 ppm, we have a multiplet that is related to 2-HG, and then a beautiful demonstration on tumors that have 2-HG, as you can see on the top line, on the top row here, where is my marker here, and not in a normal appearing white matter in the contralateral hemisphere, or I'm sorry, yes, here, and or in normal appearing white matter in a healthy subject. One should not be excused if we don't see the results of the competition. So Cheng Ho Che in UT Southwestern came up with a kind of a slightly different type of editing, if you want, of optimizing the TE1 and TE2 of a press sequence to kind of enhance the signal from 2-HG. And again, you can see here in the decomposition to the various components of the basis set in areas or in tumors where one would expect to see the 2-HG. Again, the peak in 4 ppm here appears, and here appears, and here appears, and it is completely flat where one would not expect it to be present. So this is a case in which the bench, if you want, was a phantom or a method development, and then the bedside was the application to actual tumors. And one would not, be complete if we would not close it. I'm sorry, that is actually how to make it even closer to the bedside. So here is the work from Adam Barrington and the group of Uzai about the improvement in spectral characterization of the 2-HG peaks when we go from 3 Tesla to 7 Tesla. And you can see that, for example, in terms of the variance across 10 subjects of the 2-HG characterization, we are really getting to a point where the variance is relatively small, and it's quite different from the 3 Tesla, in which there is still quite a bit of spread. So one actually should say here, well, what is exactly bedside? Bedside is where the availability of scanner is. For example, 3T is more available, or 7 Tesla, which maybe gives us better results in terms of per subject characterization. But on the other hand, it is a less available type of scanner that not every patient can jump into. But this is a beautiful result, and again, a kind of a nice progression from the initial discovery. I just show here also some preclinical results from Cheng Ho Choi group, just to kind of wrap up the story, that there are also preclinical results that can be also correlated or put side by side with histology. But I should say this is a little bit too little too late in terms of how much impact that can have on the development of the method. And it's a pity, because there was a missed opportunity thing here to have side by side animal research and human research on the same type of tumor, and to get complementary information. So again, one should redefine what bench work means in this case. which includes the development of adequate pulse sequences and maximizing the detectability of this particular marker, and phantom and simulation work that support the human data. And in this case, we sort of bypassed the preclinical work, and I think it's a missed opportunity, which again, it's a lesson learned, I think, for us to be a little bit more communicative across groups that do research in this type of topics. And the other thing is really to emphasize the rigor in which we perform our experiment. It's very, very important in this kind of relatively abstract things like spectra, that the experts should be very careful about delivering always the highest quality spectra possible, because if we don't do so, then the level of faith that other people from outside the field in what we deliver will decrease. And here I'm jumping to my third and last example, which is my kind of home turf, in which I'll talk to you a little bit about the development of clinical, I should say, or application of diffusion-weighted MRS to pathology in animal models as well as in humans. And just to show you again how information, when, let's say, a successful such network of human and animal research happens, then how the methodologies flow and how the information flows and how we can build up on results and on theory versus eventual results. I have to say here that I was thinking in the beginning to have here another successful example, which is the example of deuterium metabolic imaging, which I think is a fantastic example of how the preclinical and clinical work really work together to create what is now such a hot topic and such a beautiful method. But then I saw that Janine is giving a talk about it. Why do I need to labor? There will be a beautiful talk being given afterwards. Very briefly, we know that that MRS, one of the powers of MRS, is to be, if not cell-specific, then at least cell-preferential. Then we have metabolites that prefer to be in specific cell types. And those cell types, when there is pathology, change not only in terms of the concentration of their neurometabolites, but also in their morphology or in their cytomorphology. And some examples are, for example, neurons. In the case of Alzheimer's disease, one major change is, of course, the breakdown of the microtubules and neurofilaments and the creation of neurofibrillary tangles of hyperphosphorylated tau protein. So this is a major change within neurons that is specific to neurons, but does not happen in glia, in Alzheimer. In other diseases like PSP, actually, there are tangles also in glia, but in AD, it's specific to neurons. And we know that the immune response in the brain that consists mostly of microglia, but one should include also astrocytes, even though the primary role is not necessarily to be part of the immune system, but they also have or are part of the innate immune system in the brain. When they are activated, meaning react to some sort of a trigger, a pathological trigger, external or internal, their morphology changes. And in the case of microglia, the ramifications are gone, and we eventually end up in fully activated microglia that is a kind of amoeboid shape. In the case of astrocytic activation, there is also a swelling of the cell body and shortening of the processes. So there is a process that affects the cell shape. And if we build on that and create a hypothesis, then we can say that if we are now hypothesizing and working towards measuring the diffusion properties of those metabolites, and let's say that we are able to do so and come up with apparent diffusion coefficients of metabolites, then those would be modulated by those cytomorphological changes. For example, one can hypothesize that NFTs in AD might decrease the diffusivity of intraneuronal metabolites, or conversely, activation of a glial cell may increase the diffusivity of glial metabolites. This is a hypothesis, but this hypothesis that goes out to the world, and then people like me, like Julien, like Roland sitting behind there, and other people are taking this message and, okay, let's try to go and apply it to disease. For example, work from Julien Valette and Clémence Mieux in the Mersenne lab in the CEA. Beautiful work in which they injected into a group of rats a substance called CNTF, or ciliary neurotrophic factor. And this is a substance that is known to activate specifically astrocytes. And then they perform a set of diffusion-weighted spectroscopic measurements and look at the decay of the signal of each and every metabolite of interest as a function of the diffusion weighting or the B value, and look which of those metabolites has its diffusion properties changed the most. And the one that changed the most, or actually the one that only gives the only detectable change in diffusivity, is myoinositol, which goes back to what Steve told us earlier, is that myoinositol is suspected to be an intra-astrocytic or an astrocytic metabolite. And when one calculates the diffusion coefficients as a function of diffusion time, then again, almost at every diffusion time that was chosen here with a moderate B value of about 2000 or 3000, I don't remember properly, but there is a significant difference in the diffusion coefficient of the control group versus the CNTF group only in myoinositol and not in choline, which is kind of surprising in that respect. Concomitantly, almost, another group in Paris with another person that is sitting right here, Francesca Branzoli and Guglielmo Genovese, who is also here, took upon themselves to take it to the next stage and actually go to the real physiological property that is of interest to us in terms of the pathology, which is neuroinflammation. And neuroinflammation is something that is a hot topic in neurodegenerative diseases, many of them, in MS, in Alzheimer's disease, in Huntington disease, and others. And here it is let's say, induced model that also generates neuroinflammation, primarily induces demyelination. This is the cuprizone model in which rodents are being fed with cuprizone, which induces a temporary and reversible demyelination, which can be then recovered when the feed is going back to normal. And again, the same type of experiment. In this case, we're looking at 4 metabolites, the neuronal metabolite N-acetyl aspartate together with NAAG. Again, as a function of B value, the decaying signal, absolutely no difference in demyelinated parts of the corpus callosum of the rodent. That means that, well, if there is any change in the axons in this region, this is not something that we can detect with diffusion-weighted MRS. But as we move to be more and more, let's say, glial, if you want, here you have the creatine, already start to detect some difference, but it's not much, definitely not statistically significant. But choline starts getting quite significant, and myoinositol becomes very, very significant across the B values. And when you calculate the ADC, then you can see that the choline actually changed dramatically its ADC, the myoinositol changed dramatically its ADC, while NAA did not change at all. So here we have an additional layer. We have change in the diffusion coefficient of myoinositol, meaning maybe astrocytes again. But here we have an additional metabolite that did not change in the astrocytic activation, but changes with neuroinflammation. So we suspect that the additional change that we see in the diffusivity of choline may be related to what we see as activation of microglia. We don't have a full support for that, but it might be an explanation for what we see. Interestingly, you can see that concomitantly one can measure the concentration of those metabolites. And the changes here are actually in NAA changes dramatically, but the myoinositol concentration doesn't change. So we see that there are 2 at the price of 1, if you want, that you can get information from both the concentration and the diffusion that would be complementary to one another and will probably signify different aspects of the pathology as we are looking at it. Since we're talking about preclinical or animal experiment, one can do histology, and what they did is indeed histology and stained for both astrocytes with GFAP and for microglia with IBA1, and excellent correlations, by the way, between the staining and the changes in ADC for both choline and myoinositol. So these are also supported by immunohistochemistry. Meanwhile, back at the ranch, there is Itamar and other people that are doing this type of experiments, but only in humans. So one of the first works that we did was with a cohort of subjects with neuropsychiatric lupus. Lupus in general is an autoimmune disease. The neuropsychiatric aspect of it is when the autoantibodies that act against our own body are reaching the brain. And this is a very devastating aspect of lupus, of systemic lupus erythematosus, where subjects wake up in the morning, in the light case with headaches, but all the way to psychotic breaks. So there is a huge range of symptomatics there, and they're quite helpless. But funnily, or luckily, I should say, with treatments that are addressing the right phenotype of disease, either the ischemic phenotype or the inflammatory phenotype, one can actually make them better and to make the neuropsychiatric symptoms recede. And here we looked at diffusivity of metabolites in normal appearing white matter part of the corpus callosum. And again, by looking at the diffusivity of the NAA from healthy controls to patients with lupus, but without neuropsychiatric symptoms, to those with neuropsychiatric symptoms, in the NAA diffusivity, there was absolutely no difference. But in the choline and in the creatine, interestingly, we saw a kind of gradual increase in diffusivity in these groups. What does the creatine have to do here? Well, we also have a suspicion that is not substantiated, but has support from an older study of ours in which we did functional diffusion-weighted MRS in the visual cortex, that enhanced metabolism or hypermetabolism changes in a non-explained yet way the diffusion properties of creatine. This is also supported by findings from the group in Paris in MS, where in areas where it's suspected with inflammation, there is also an increase in the diffusivity of creatine. The latest, a dernier cri in showing what happens in neuroinflammation to the diffusion of metabolites, is an experiment actually that I participated in, but did not initiate, in Brighton and Sussex Medical School when Mara Cerciniani was still the center director there. And the idea came from Neil Harrison, who is a psychiatrist and a physiologist, who had an idea that a model for inducing systemic inflammation in animals and in humans is to supply them with lipopolysaccharides, that is the coating of a specific type of bacteria. The body reacts to the LPS as if there was an invasion of bacteria, so fever and feeling quite lousy for about 7 hours, and then all of a sudden feeling better because it has been cleared up. And the hypothesis was that if you induce systemic inflammation, there might be a gateway to neuroinflammation for a temporary time, especially through the thalamus. Don't ask me exactly how, because that's the part where I am very weak at, but not so weak at generating experiments. So what we did is we did the diffusion-weighted MRS, in which we put a volume on just parietal white matter and in the thalamus. And in the thalamus, we showed a very significant increase in a small cohort of subjects of the diffusivity of choline that may suggest that there is indeed a reaction of the immune system in the brain to this infusion of LPS. I want to conclude the talk with the last slide, in which I showed just data, by paying tribute to our friends that I think are online. That's Shi Chen and Fei Du and Dost Ongur from McLean Hospital, that actually use diffusion-weighted MRS to look at patients with first episode schizophrenia, and to show that from first episode schizophrenia, the ADC of choline is elevated compared to what one would measure in consecutive years, and the NAA, the ADC of the NAA, which is at normal levels at the first episode, actually increases. So that's a very interesting finding that again shows how a group absorbed methodology and applied them to their own questions. So the take-home message is that the success, and I really call it a success in the sense that we don't have it diagnosing patients at any time soon, but it is a program that started, I would say, from being a very esoteric measurement, and now it is becoming more of interest to the clinical community that comes to us to ask, well, can you give us your patch? Can you give us your pipelines, etc., and so on and so forth. So the success depends on generating those very highly specialized pipelines that go from the preclinical to the tissue to the human models, and importantly, to have a completely open approach where data and methods flow across groups, very tight quality control over the data, and importantly, providing an entire easy-to-use pipelines to interested researchers. And the challenges are, of course, to bring the methodology to commercial platforms, and that's a kind of a difficult point to make methods pass across platforms. and to actually convince the vendors to incorporate them in the standard software, for example, and of course to expand the circle of users. So just to summarize everything, there is no directionality in what we do, but this is just fine. We're doing okay. And we have lots of components in this network, and each and every component has the possibility of contributing to the success of preclinical to clinical to tissue model, this kind of cross-model program that I just mentioned yesterday. And I think that the main point is that really we as methodology people should educate ourselves in what the community, the user community needs, and educating the community about what we can deliver and what we cannot deliver. And this is extremely important, I think. And last comment of all is that MRS is not a silver bullet, and one should always think a little bit kind of multimodally. If you can add additional data to support, to make the accuracy or the precision higher just by additional data, even on the clinical level, it is, I think, an important thing to do. So thank you very much. And I think everything is open for discussion. So thanks so much.
Thanks a lot, Itamar, for this vintage performance. And now the floor is open to questions. And I will pass the microphone around as quickly as possible to adopt your new discussion format.
Thanks, Dr. Ronan. This is, I was jumping out of my seat through much of this talk, as you probably saw, because as you call it, the cross-model program is exactly where I, for one, and many others, hopefully, want to see this community being involved in. I have a question that it specifically may not be that interesting, but it is a question that I so frequently have during these kinds of discussions that I'd also like for you to maybe, for the group to discuss it more broadly. You mentioned the choline, a possibility that the, I think it was an increase in choline, you gave an example of some physiologic or pathophysiology reason why it may be changing. But are there other possibilities that might be related to the physics of MRS and the way we're measuring that could be acknowledged and or ruled out? Could you discuss that? Does that make sense? Okay.
Yeah, I mean, first of all, thank you, Jody.
That's a question I can't answer.
No, I totally see your question. I will be glad to start the discussion about it, but again, I'm very glad to have additional inputs to that. I think the general question is, how do we rule out other possible mechanisms in a specific finding that we think that goes in a specific direction? And my simple answer to that, to this very not simple question, is to try to educate ourselves as much as possible with respect to everything that goes from the measurement to the system in which we're looking at, to the physics that we're looking at. And I suspect that, for example, in the particular question that you asked, for example, there are a host of alternative explanations that we are simply not aware of. Like, for example, the fact that, for example, let's say that the increase in the diffusivity of metabolites is linked with some sort of process that happens in astrocytes. But astrocytes have lots of roles, right? They're part of the blood-brain barrier, and they're part of the immune system, and everything about them can be modulated by many different reasons. So we have to systematically, of course, either find a way to exclude those reasons from being the explanation for what we see, or to include them as possible explanations as well. So this is why I think, talking with certainty, especially, and this is a pitfall, I think, in human MR, and I want to go back to things that Steve mentioned earlier. For example, one thing that we're taking completely for granted is this, and you see it in many MRS papers, is the subdivision of metabolites with respect to its population in different cell types. what we know is a lot of what we know comes from Steve's earlier studies in cell cultures and in some animal models where we can do some sort of quantification. But I think that we were, how shall I put it, hasty in jumping into using those numbers as truisms in everything that we say about MRS in humans. We do not consider many things that are, for example, very important to consider when we talk about concentrations, but also, let's say, more complex quantities that are related to, for example, what is the cell density? What is the size of the cell, and actually how much of this particular metabolite exists in the microglial population within 1 cc of tissue, which includes not just its concentration in the cells, but how many cells are there and how they are distributed across the brain, right? We really don't know this. And I think that what I saw now, for example, with the attempt at explaining, for example, the increase in glutamate with the vesicular release and so on, there is already, you know, people are catching back into those kind of basic questions that I think we all should actually go and try to answer. I hope this answers somehow what you asked, but I'm glad to have other people bring their thoughts as well.
Can this be a quick one, please? We are
Yeah,
way over time.
Yeah, I just wanted to have a comment for the, you mentioned about this missed opportunity to do preclinical, clinical together, or preclinical prior to the clinical for IDH glioma. I think the reason for that was not because people didn't want to do it, because the model, it was very hard to have a mouse model that was orthotopic xenograft human IDH glioma cells to grow. So it took a couple of years until there was a mouse model available for that. So there was no opportunity but to do it first in humans.
Can I go?
That happens with many diseases.
But can I ask you a question back? So to be blunt, we know that many diseases don't actually have animal models. Okay, so for example, for neuropsychiatric lupus, there is a model, but the model is very weak. Or many, let's say, of the rare metabolic diseases, there are models for the disease itself, but the way that it expresses itself eventually in the brain, it's not there. Jody, you may talk about, you know, psychiatric models or schizophrenia models for rats or mice or whatever. I'm not sure we can do, you know, a proper interview with the rat to get or with the mouse to get exactly the same kind of symptomatics that we see in humans. So is a preclinical model really always necessary?
So if I get the chance for a second question is, like, what is the biological explanation for this opposite changes in choline and NADH in schizophrenia? Like the last slide that you had. Like you showed that the choline and NADH changes in time in opposite direction. What would be the biological interpretation or
the biological interpretation in general would be that the episode or again, I will not replace anything that, of course, Dost and his people can explain probably much better, but in short, and that's not only in schizophrenia, but also in other neurodegenerative diseases. For example, even in Alzheimer's, it is suspected that the initial stage includes a neuroinflammatory response, which is responsible for the increase in the ADC of choline, and that the neurodegeneration that gradually takes over later will eventually be the factor that affects the diffusivity of the intraneuronal metabolite. So the timeline is such that in first episode schizophrenia, the main event is an inflammatory event, whether the neurodegeneration driven either by the disease or by the medication or by both will be a progression later in the timeline. That's my understanding.
Thank you very much. I hate to do this, but we now need to limit the discussion or postpone it to the lunch break.
Sounds good.
Then we have to move on to the next talk. Let's thank the speaker again.
Thank you.
Oh, yeah. That is. Thank you. Thank you. And now we welcome Martin Prischap from the University of Vienna, who will take us on a journey outside of the brain. So his talk is entitled, hasn't been shown up here, Methods for Non-Brain Clinical MRS. And we look very much forward to your talk, Martin. Thank you.
Thank you very much for the introduction. Thank you very much, Christina, for inviting me to talk here. And also, we have some discussions with her and with Vera about what should I talk about, that not to give a big overlap between my and Vera's talk. So I'll try to get at least a couple of points on translational things on non-brain MRS. We are in a, there are a lot of applications, a lot of methods here, so I will not cover everything. I'm glad that that probably you can recall the lectures before, and I can build up on some of the artifacts and on some of the quantification things have been said and on translational. But so let's start with what the MRS outside the brain is used mostly for. As been said here, oncology is the bread of spectroscopists that has been used or applied or attempted in outside the brain. But the main part of MR spectroscopy outside of brain has been applied for metabolic studies. So while in oncology, it's more kind of lesion detection, characterization, and characterization of the treatment response, in metabolism, these are the questions related to diabetology, sports physiology, tissue degeneration, or characterization of diffuse disease. And here below are the organs listed, which were mostly attempted or which are examined by MRS. The oncology, I'll tackle a little bit of breast and prostate MRS, and in metabolism, I'll talk more about skeletal muscle, adipose tissue, liver, and heart. And also the spectrum of nuclei we are using here, it's quite broad. My talk will focus mostly on proton, a little bit of natural abundance carbon studies and phosphorus studies. We will hear about deuterium spectroscopy tomorrow. We will hear about hyperpolarized carbon MRS tomorrow. So that is a big help for me. So if we go for outside of brain, this kind of the strategy of whole experiment starts already with not only with data processing or acquisition strategy and data processing, but what we need to think about is what kind of coil are we going to use. They are mostly not kind of out of shelf or not bought at the first tranche of buying of the experiment of the experimental setup, and also the positioning of the patients. And all of these points like nuclei, RF coils, positioning, acquisition strategy, and data processing are kind of interconnected. And also there is no streamline or no linear way how to think, because sometimes you start with nuclei, have an RF coil, and then you say, okay, but I cannot do this acquisition with this. So I need to adjust the coil to my acquisition strategy or data processing and so on. So if we look at the coil, what we are using is a broad spectrum of coils here, starting with surface transmitting receiving coils with rigid, shaped, or flexible coils. Then some organ-specific coils, like for the breast spectroscopy or breast imaging, and endorectal coils for prostate, and combination of volume or whole body transmitting coils and surface receiver coils as here, or whole body volume TX, RX coils. And all these, again, in all possible combination of proton, deuterium, carbon, and phosphorus, being it double-tuned or some triple-tuned coils used as well. When we think about localization, even so in the brain, you are used for single voxel or CSI, but outside of brain, and if we go for carbon or phosphorus studies, even non-localized or some 1D slab localized sequences, acquisitions are good enough or are satisfying for the questions here. If we tackle our questions with single voxel spectroscopy, we should not forget about adjusting the T of the sequence to T2 of metabolites we are looking at, about partial volume effects, chemical shift displacement effects, and so on. And sometimes, especially for phosphorus spectroscopy, ICIS localization is used a lot. For CSI, it started, of course, with the press or steam box, but also FID-based sequences, again, especially for carbon or phosphorus, are used to. And also the combination of the excitation volume and the sensitivity of the surface coil is a lot of times used for a kind of combined localization. Outside of the brain, some of the movement of the patients we cannot restrict. I mean, we were talking about the padding yesterday here. I mean, my favorite way of padding of extremities are the sandbags. They are very comfortable, and this kind of restraining of the patient is also very good accepted. But if we look at the spectroscopy of the heart or of the liver or in wherever in abdomen, we need to suppress the breath movement as well, and also to suppress the influence of heart movement. So acquisition during a breath hold or during multiple breath hold are applied. Also, we do trigger our acquisitions with breathing, with ECG, or use some kind of navigators. Or the movement can be also suppressed in post-processing of single FIDs and individual phasing, frequency alignment, and discarding of corrupt signals, as has been done in some liver studies and also in gallbladder studies, for example. So for the spectral fitting, we have seen also beautiful lectures here. We use everything basically the same as in the brain. Frequency domain, especially for protons, and combinations or different approaches for frequency and time domain for phosphorus and carbon. What we should not forget, there are unique and tissue-specific spectral pattern. We are not in the brain. Some of the vendors supply you with kind of basis sets or prior knowledge for muscle or for the liver, which includes also, for example, lipid resonances. But some of the vendors do not. They supply only a sequence-specific prior knowledge for the fitting, and so you end up measuring the liver, and N-acetyl aspartate shows up in your spectrum, in your process spectrum, and then you get calls from radiologists. How comes? What is this? I cannot see the lipids in the liver. Yeah, okay. So that's we should not forget about this. Yes? So that these tissue-specific spectral patterns are necessary and should be applied. For the quantification, we had also a very nice talk here. So the internal reference, especially in proton for water and creatine, or in phosphorus case, beta-ATP is a lot of time used. We use a phantom replacement technique in phosphorus and carbon spectroscopy. I'll call it a rather phantom replacement than the external phantom or external reference. And also, we start with relative units of quantification. I'll tackle that at breast and prostate cases, and for example, in hyperpolarization and phosphorus as well. And what is necessary, of course, if you go and give some number, either you know T1 and T2 and do the corrections, or you explicitly say that it was not corrected and that your numbers are kind of arbitrary units as well. The standardization of experiments is necessary also, of course, here. And we had two nice consensus papers in the special issue of NMR and Biomedicine, one of the proton MRS in the muscle and one of the phosphorus MRS in the muscle, which summarized all the technical aspects and brought the points which should be followed. So let's come to the practical examples to show you some spectra. So I'll start with oncology, and the attempts to characterize the lesions of in the breast with the spectroscopy came in early 2000s or before, and what you see here that you are not trying to suppress one peak of water and have whole spectrum of peaks, but more or less what you are trying is to suppress water and fat and to get a decent signal of choline. And the attempts of single voxel spectroscopy or press box CSI were done. And here you see the choline peak inside the lesion and kind of no choline peak outside. And you can build beautiful CSI maps. But the point is that one of the limitations was that the absence of kind of absolute quantification, the total choline SNR was used in these spectroscopy attempts. Another point is one of the partners in crime is here in the audience, that the other MRI techniques showed much better performance regarding the classification of the lesions and being it diffusion, so measuring of ADC of the lesions or combination of ADC and dynamic contrast enhanced MRI showed much better in this respect. So beautiful technique, a lot of work invested into it, but at the end it turned out that the spectroscopy is not bringing that what we would like to. Yeah. Similar case, spectroscopy of the prostate. Also, the heyday of it, maybe 15, 20 years ago. Here again, a little bit different kind of quantification is used. The citrate peak you would not find in the brain. So again, a tissue-specific spectral pattern. The citrate is a marker of healthy prostate tissue, and the choline, of course, the peak or the marker of the malignancy. But especially at 1.5 Tesla or even at 3 Tesla, the resolution of choline and creatine at the prostate could not be achieved perfectly. So what was used was a choline plus creatine to citrate ratio as a marker or quality or as a tissue distinction marker. Here you see a spectral map on the console and also the CSI map on the console. What was one of the biggest problem here in the prostate was the quality of the spectra. We have these talks yesterday. You have seen. I mean, back in 2010 or so, our colleagues used the relative CRBs as a quality measure and tried to set up a kind of easy going or easy accessible measurement of or display of spectral quality, kind of shading the spectral map. Here you have the red voxels, which are obviously myelin, so the choline is high, and the spectrum is okay. And here the red spectra, which are where the choline was quantified high, but the spectrum was not taken as a good one, similar to the green and areas. Here in prostate again, very demanding examination, very demanding processing. A lot of work has been done, but again, the ADCs and DC MRI, the combination of it showed better clinical performance than the spectroscopy. If we if we turn to the metabolic part of my talk, why it's important, why the spectroscopy, or where it's going, it's the glucose and it's fat. So the lipids and fat. So the artifact of yesterday is being measured here as a measure of fat accumulation, lipid accumulation, but also not only accumulation, but also a quality of the fat profiling. By building a ratio of double bond resonance to a methylene group resonance, we can see differences in a fat profile of visceral adipose tissue of lean and obese patients. So these obese patients have not only more visceral fat, which is this belly fat we don't want to have, but also the quality of the fat is more saturated. So, and if we turn take this is a short echo single voxel acquisition, and by, for example, by long echo single voxel acquisition, we can discern even the omega-3 lipids out of the saturated lipids by looking at only the sub-region of this methyl peak. Very first applications of proton spectroscopy, even in the clinics or up to the clinics, were the measurement of the fat in the liver. Here you see the strong lipid signal from a liver which is obviously full of lipid vesicles, lipid tissue full of vesicles, and here very weak lipid signal from a hepatic tissue which is very healthy. And there are several papers showing that MR spectroscopy can be used for grading the steatosis, and also can be used for grading of steatosis. Skip this one, but this for the methodology. The importance of T2 correction, and especially in the liver for individual T2 corrections of the spectra, pointed out that it's necessary to have a multi-echo acquisition. Especially in the liver, you have a lot of times the fat influences the water relaxation time, and also the iron deposition influences the T2 relaxation times. And early in 2000 years, several groups came out with kind of multi-echo acquisition, spreading the echoes from something like 12 to 70 to 80 milliseconds, 5 echo times, and showed that single transient of MR spectrum, proton's MR spectrum, is capable of quantifying the liver fat enough. And this was a first product tool I saw from the vendor, giving it as a quantitative product on spectroscopy. So here you see there was a 1, 2, 3, 4, 5 echoes acquired during one breath hold, and that led to graphical display and numerical display of fat accumulation in the liver. Nevertheless, This is a single voxel acquisition, and by the time in early 2000, the multi-echo Dixon acquisition suffered from some artifacts or drawbacks, especially in the lower fat acquisition area, so like 1 to 5%. They were not accurate enough, but it evolved. So I would say that the spectroscopy here again pushed the imaging to perform better, especially in a very critical areas of telling if it is pathological accumulation of fat or not. And nowadays, especially in clinical application, the Dixon prevails. So the vendors are selling very nice packages, which are even segmenting the liver and giving you a value of liver fat over the whole volume. But spectroscopy can do better again. Not only quantifying the large and strong methylene peak, but also quantifying small peaks, and with some special, especially so we can quantify olefinic lipid resonances at 5.2, 5.3 ppm double chain FA. These are the data from 7 Tesla from our lab, but I think tomorrow you will see very nice 3 Tesla data from Vera. Bringing the point also the connects to the physiology.
I've been sorry to interrupt, but can you wrap up maybe within the next 100 minutes?
Yeah.
We already. Sorry, I didn't want to pose such a problem for you now.
Okay. Yeah, so let's come to one of the so a lot of you know about the IMCL story, the intramuscular lipid story, which was started here in Switzerland and then taken over. And again, there is an artifact of the artifact. If you look at here at the spectra, so we are not looking only at one lipid peak, but 2 lipid peaks, and this one even moves around the spectrum as you position your patient in different way, or as you position your phantom in a different way. So the positioning is very important to get this very nice resolution. And here also the issue of positioning and also issue of these translational studies came into the play, where here you see the same spectra from the animal, and this spectrum is taken from the same position. These 2 spectra are taken from the same position in the red, with the difference of the volume. So this is from a lower volume and this is from a larger volume, and this very nicely shows you the heterogeneity of this extramuscular peak, which is kind of this marbling of the steak. So very heterogeneous distribution. And if you focus very well in the muscle, then you can take only the intramuscular lipids. The second one, I wanted to show you again about the positioning, how the coil works. So this is a simple surface coil, and the coil and the images and spectra taken with a Helmholtz coil, adjusted specially for this application. You see like doubled increase of signal to noise, and also the Helmholtz coil led the inventors to the point that the positioning device within the coil, it's a red calf, needs to be done professionally, not only with some padding and every time a little bit different, and designed the coil with the positioning especially for this issue. So I had a little bit more prepared for this I'm sorry now to be a little bit victim of what we have before. What would be one of the good point? I tackled the phosphorus spectroscopy, where we have this consensus paper. Here again, the unlocalized spectroscopy can be performed in many clinical scanners have been done in 1985, I think, started with. We can draw a lot of useful physiological information out of it during exercise challenge. But what is important here, that the localization plays a role also in discerning the muscles which are working and which are not. So the unlocalized spectroscopy works fine, but localized gives you much more specific information, being it single voxel or a slab localization. And also what is important at this translational point, that what we can do now is go for multimodal interleaving, measuring perfusion and metabolism at the same time, or measuring acetylcarnitine and phosphorus metabolism, so two proton and phosphorus spectroscopy at the same time, or measuring the lactate, for example, with double quantum filtering and the phosphorus spectroscopy in the same time. Animal work has a special things here, special restraints, because we cannot tell the rat, the animal, to treat the pedal, so we need to force it somehow, and the animal is anesthesia. Here, very nice work of Marseille group, and all the things. And the quantification is important, but this we can talk about during the break. So we need adjusted to organ, the experimental setup needs to be adjusted to organ and research question. We can quantify energy turnover, lipids, and glucose metabolism, and it's the monitoring of tissue-specific and dynamic responses to metabolic or exercise challenges important and possible with non-brain MRS. Thank you very much.
Martin, thank you a lot. One quick burning question I will admit, because it's the first talk about non-brain MRS. If there's not something, I would really encourage people, ask Martin during the break or after the program, because he had prepared so much more interesting material. Just unfortunately, we are running out of time. So we will move on, and I will hand on the moderation to my co-moderator. Thanks again to the speaker.
Hello again, and we have to move on, and it's a time for the new scientists. Then let's turn the floor over to the new scientists. I would like to invite Julian Levenbach from the Maastricht University. The title of his work is Alpha-ATP suppression in the phosphorus MRS spectroscopy by homonuclear bird editing. An approach to quantify NADH and NAD plus at 3 Tesla in a skeletal muscle. Floor is yours.
Thank you very much for the nice introduction. I'm happy to be here. Thanks also for the committee for inviting me. And indeed, today I want to talk about NAD quantification, also a non-brain spectroscopy topic. And first of all, a short introduction. We had a presentation about NAD quantification yesterday already. So you are familiar with the idea that NAD plus and NADH form a redox couple and are quite important for the energy metabolism. And it is so far known that athletes or people that are metabolically healthy have a higher NAD plus to NADH ratio than metabolically impaired or older people. And this has been shown earlier as well. The thing is, though, NAD so far in the muscle was mostly quantified by taking muscle biopsies, so invasively. It was kind of unpleasant and a bit risky. So the idea was, how could we do this with MRS? And we came up with an idea to measure it in phosphor MRS, as NAD plus and NADH are visible there. But how to do this? So our objective first was to develop a sequence that is able to measure NAD+, or at least a protocol that can do so. Then we wanted to apply this method in a young, healthy population and test it on an ischemia protocol to measure changes in NAD+ and NADH. Then we wanted to look at differences between groups. For that, we had older trained adults and older sedentary adults. So we had a difference in training status. So first of all, why is it so difficult to see NAD in a phosphorus MRS? It's a bit hidden. I admit it's not the best quality FID that is here, but for this purpose, it will do. It sees that NADH and NAD+ are overlapping with α-ATP at 3 Tesla in phosphorus MRS, and it's quite hard to just disentangle these two. So what we want to do with our new sequence is we want to suppress α-ATP and get rid of it. So just see the NAD peaks. How can we do this? Well, spectral editing is the answer. As an approach for spectral editing, we were going for the basics of J-coupling, as α-ATP is a nicely coupled signal in phosphor. It's coupled to beta-ATP. Over an echo time, it will change its phase relative to uncoupled peaks in the spectrum. And for that, we have developed a double echo sequence that, based on the inverse J-coupling constant of alpha-ATP, basically selects alpha-ATP by waiting until the ATP resonance has acquired a phase of 90 degrees in respect to the NADH and NAD+ peaks in the spectrum, or in the scanner actually. Then flips it about the arbitrary axis at which the ATP peaks are along to leave these in the XY plane. A spoiler gradient dephases these signals, and in the subsequent FID, we are only left with signal without alpha-ATP in it, hopefully. So what we've done then is we went off from the drawing board, we programmed the sequence and tested that in a phantom. The phantom was composed of ATP and NAD+, and a PI peak, nicely visible to the left. And we've tested it. So, and indeed it worked. We suppressed alpha-ATP by about 85 to 87%, and had a signal loss of about 23% on the NAD+ resonance. Nice and well. Peak is a bit better visible. So what do we do now? From this point, we went into an in vivo situation. So we measured in the lower legs of human indeed, young, healthy participants. They were doing a MRS at rest, during which we did a power optimization and some other things that will come later. And then we also started an ischemia protocol, whereas ischemia would basically, as we expected, rise the amount of NADH, as the electron transport chain without oxygen supply from the blood would be blocked, and NAD+ could not be oxidized back to NADH could not be oxidized back to NAD+, so it would accumulate. Our protocol consisted of sequence optimization, as we had block pulses in the sequence. These needed to be power adjusted, so we just checked where the at which apparent flip angle the PCR peak in our spectrum was maximum, used that power setting. After that, we performed a regular FID for a reference scan. Basically, you have an idea of the quality of the spectra. We then performed the baseline scan with the editing at rest to determine resting NAD levels and NADH levels. And then we did inflate a cuff that was placed around the upper leg of the participants before they went into the scanner. Inflated this cuff by 30 millimeters Hg above their systolic blood pressure to induce ischemia. And we measured then during about 9 minutes to see what changes happen. What we found was that in this setting, the NADH indeed increased, as we can see here in the baseline fit and in the ischemic fit. The fitting script, just briefly mentioned, also mention of time, is in-house developed, and it is a MATLAB script or a MATLAB algorithm that allows for fitting of frequency shift, zero order phase, implementing of prior knowledge of the peaks, such as T2s, J-coupling patterns, and phase artifacts due to editing. So it was quite flexible for us to use. And it achieved Kramer-Rao lower bounds of 3.9% for the NAD+ resonance and 9.76% for the NADH resonance. So we were quite happy with this, indeed. And the clinical results from our study showed us the expected changes. The NAD+ levels during ischemia were dropping, whereas the NADH signal would rise, which would also then, of course, reflect into a lowered NAD+ to NADH ratio. In this case, we have reported arbitrary units. As for absolute quantification, you would have technically do a T2 correction due to T2 loss from the editing, but the T2 of NAD and NADH in the skeletal muscle was not known yet, so that was a bit difficult. But as we had an intra-individual comparison in an acute situation, the changes were still shown, so not necessarily in absolute quantification. Going over to the situation where we checked between the trained and sedentary groups, our marker there during screening was that the sedentary groups had no more than 1 hour of structured exercise per week, and the trained groups would engage more than 3 hours a week in sports. Next to that, we also performed a PCR recovery experiment to basically check how trained they actually are, because PCR recovery, as Martin also touched upon, shows us the oxidative capacity of skeletal muscle and is an indicator for fitness, oxidative capacity in there. Next to that, we also performed the resting state MRS as shown earlier with the editing to suppress α-ATP to get then their NAD+ and NADH levels. Again, the reference, FID, and sequence optimization were performed, power optimization, followed by the 2 experiments. Resulting from this, we saw that indeed the screening showed, or from the sportive and active people that were giving them the screening, indeed had a better oxidative capacity and a better PCR recovery rate than the sedentary ones. And here we also see that the NADH levels in the sedentary ones are higher than in the active ones, as well as we see higher NAD+ levels in trained versus sedentary. So what we expected actually also was reported here. The only thing is that the total amount of NADH did not differ between trained or untrained. So it seems to be not much of an effect there. So coming to the last points, what we've managed to do so far is we have developed a phosphorus MRS technique that can show us NAD+ and NADH, and also determine the ratio between NAD+ and NADH. We can pick up the expected changes during ischemia. We can show that there are differences in the composition of NAD+ and NADH in the ratio between trained and sedentary in older adults. What has not yet really worked is visually separating NADH and NAD+ from each other at 3 Tesla, as the spectral resolution is just not good enough. But maybe to make the quantification even more robust, it would be an idea to increase the amount of quiet spectra, to increase SNR, but also to suppress α-ATP a bit more. We're not quite sure why there's so much residual α-ATP. So the idea is that there could be something such as biological flux that would just make new α-ATP, or new ATP that will not be edited, but just appear in the spectrum in the FAD, or that maybe also, because we saw in the phantom quite a nice result compared to in vivo, that the B1 inhomogeneity is a bit bigger there, of course, in vivo than in the phantom. So we should look into that a bit more. This is also stated as questions, so if there are any suggestions, I'm happy to hear them. And with that, I thank you for listening. It's going a bit faster, but just in time. Thanks everyone who was involved. Thank my supervisor and colleagues. And yeah, that's it.
Thank you so much, Julian.
Thank you.
Especially for timing, proper timing. So we have a time for question or comments. Is there any? Yeah, please.
Thanks. Yeah, I love seeing phosphorus spectra. Reminds me of my youth. Do you think you're measuring, well, do you know whether you're measuring mitochondrial or cytosolic NAD?
Yeah, we're assuming it's more the cytosolic one, but actually a combination of both, as we cannot quite see the distinction between the two. There is a slightly different pH level in mitochondria, so you might expect separated pool between the two visible in the spectra, but we have not seen it in the data so far.
Because I mean, the NAD to NADH ratios are different in mitochondria and cytosols. So
that is a very good question. It should be higher in mitochondria during oxidation, indeed. In the cytosol, oh, I have to give that a good think.
yeah, I think it complicates the interpretation.
Yes, indeed.
Yeah. And so can you measure? Can you measure both ATP and PI?
Yes.
In your spectrum. Yeah.
Mhm.
Because the ATPase should be in equilibrium with the cytosolic redox potential.
Yeah.
So your ATP to PI should mirror your NAD to NADH ratios. So if one, I can't remember, one goes up, the other goes down, but they should be coupled.
Yeah. I should just see if that also resides then in the editing, or if we would have to do a separate experiment with only an FID, another acquisition method.
Well, presumably you still see the beta ATP.
No.
No, okay.
That one goes away during the editing.
Yeah, well, PCR to PI would be a good secondary marker of that.
Yeah, but there you also see a reduction between the resting state and the ischemic state.
Yeah.
So that will be a good one. Thank you.
Thank you so much, Steve. If there is no any other question, we immediately thank you again, Julian, for your nice talk.
Very welcome. Thank you.
Now we can move on to the next speaker, which is Moeed Azhar, and from Cambridge University. And the title of his talk is Association of the Insulin Resistance with the Accumulation of the Saturated IMCL compared with the other source. Floor is yours, Moeed.
Thank you. Good afternoon, and I'm afraid you would be hungry, so I will take like 10-15 minutes more. So I'm talking about association of insulin resistance with accumulation of saturated IMCL and compared with other fats. So let's look at MCL or lipids again instead of discarding them. So intracellular lipids are lipids droplet located within skeletal muscles cells, and while the extracellular are the one which is outside the cells. Usually, biochemical analysis can't distinguish between intracellular lipid from extracellular lipid. On the right, you are seeing a 1H spectrum, which is individually and overall fit for CH3 and CH2 intracellular and extracellular peaks. So 1H MRS can non-invasively distinguish between IMCL and EMCL. This led to use the CH2 peak to look into or study IMCL, but CH2 peak is the composition is dependent upon the composition of IMCL. On the other hand, the CH3, which is independent of its composition. So why not look into both peaks, CH3 and CH2 peak? So previously, we have validated the compositional marker named IMCL saturation index at echo time of 35 milliseconds using press sequence. On the right below, you are seeing a panel, which is the main principle. It shows that as the degree of saturation increase, unsaturation increase, the value of the ratio changes. So on in the upper, you are seeing palmitic acid component of triglyceride molecules with theoretical ratio of 8. And with the introduction of single bond, the ratio decreases to 5.3. This has been already published in a Thekamani paper. Next step is, let's see its relation with insulin resistance. So we use this marker to show that saturation of IMCL is more closely related to the whole body insulin resistance than concentration independent composition. So you can see the table here where we have concentration on the left, concentration composition of CH2 peak, and composition adjusted quantity. So what we can see here, that the adjusted quantity is more closely related with insulin resistance in controls and healthy subjects. So what is this adjusted quantity? This is basically a linear regression through the control data points, having the slope of CH2 over CH3, having any pathophysiological meaningful measurement of this composition. Any vertical deviation from this line is measured and taken as a marker. And this is basically an adjusted value. So this is already being published in the Savage paper. So after looking into the relation, so the next logical step would be whether this marker, CH2, CH3, associated with insulin resistance is stronger than previously reported association with other ectopic or adipose tissue stores. And second, whether it remains a significant predictor when these are taken into account. So we start the study with recruiting 38 healthy females, 27 healthy males, having their age and BMI index matched. The whole body fat was measured using dual energy X-ray abdominoplasty, and while whole body insulin resistance or HOMA-IR is measured using this formula. So we start with MRS protocol, start measuring their fat contents in soleus and tibialis anterior using obtained spectrum using press sequence at time echo of 35 millisecond, water press spectrum using TR 5 second and 64 averages. While intrahepatic lipid was measured at the voxel within the posterior aspect of the right lobe of the liver using the press sequence at time echo of 35 millisecond, we obtain non-water suppressed data using TR7 and 64 averages. The data was analyzed in GMR UI and using MRS algorithm with prior knowledge. So the rest of the fat was measured using MRI protocol for abdominal, visceral, and subcutaneous adipose tissue, wet and scat. We obtained T1 weighted turbo spin echo water suppressed transaxial image at the location of L4 vertebral level, slice thickness of 10 millimeter with TR 400 millisecond, echo time of 21 millisecond to averages. Volume of the tissues were calculated by using a threshold map in combination with manual input to distinguish between wet and scat compartments, while calf intramuscular fat and subcutaneous adipose tissue was measured using obtaining an image T1 weightage transaxial slice centered to the 1H MR voxel. The regional MRI analysis was performed and analyzed. So the statistic was done in SPSS with non-normal measures were log transformed prior to the Pearson correlation analysis, and the Benjamin Hochberg adjusted p-values were used to correct to the multiple comparison. So let's move to the result. On the right, you can see the females and males with their age, BMI index, and also their whole body insulin resistance. On the right, you can see the table consists of correlation coefficient of IMCL concentration, composition, and other fat of the females, males, and combined. So if we see at this table, we can see Zolius adjusted marker has a close association with insulin resistance, also IHL, and total fat mass also have the higher correlation with the body insulin resistance. So you can see the plots here where we have on the left is insulin resistance versus Zolius adjusted marker. We can see the positive relation and also on the other side. But when we do the stepwise analysis, we find out that the predictor in the female is a wet, in the male is IHL, and in both IHL and fat mass is in female and the male. So the conclusion, as well as the accumulation of saturated IMCL, several other markers were, as previously reported, significantly associated with the whole body as insulin resistance, namely wet, IHL, calf subcutaneous adipose tissue, total fat mass, and abdominal subcutaneous adipose tissue, combined in male and female. Disentangling these relationships by stepwise regression did not reveal the accumulation of saturated IMCL to be a significant predictor of whole body insulin resistance, which implies these measures carry no more information in practice than the predictors we found, such as intrahepatic lipid. However, the performance of any marker depends on factors such as measurement precision and accuracy, biological variation and dynamic of range in addition to the underlying biology. As the dynamic range and precision of the predictor is much greater than that of the saturated IMCL marker, this negative finding does not rule out a role for saturated IMCL accumulation in the pathogenesis in insulin resistance. So in the end, acknowledgement. Thank you very much for listening.
Thank you so much, Moni. Last slide.
Please roll it.
Yes.
Yeah, just a quick question. You call this saturation index, but I mean, the main factor would be chain length. So is there the chain length is constant? Are you sure about that?
Yes, the primarily effect is basically the degree of unsaturation, but the secondary effect for sure is the chain, which we haven't calculated. So of course there is this effect, but it's not that much, it's minimal, so we have neglected it.
Maybe another question. I think you probably showed, didn't show soleus muscle, like I suppose. Your separation IMCL, IMCL probably is worse in soleus than in tibialis or gastrocnemius. Separation is good enough for this?
Yes, we are able to locate it with the pure knowledge. Yes, you're right.
Thank you so much. Is there any other comments or question? Thank you both audience and speakers for their nice talk. And so by this, we want to conclude this session. Enjoy the good discussion with the young scientists and hopefully delicious.
Okay, good afternoon, everybody. Welcome back. I'm Francesca Branzoli, and I'm very glad to be moderating this session on accuracy and rigor together with Jesse Mosso. So I'd like to introduce the first of the session. Maybe we should. How about that? Great. It seems we are ready. So the first speaker of the session is Gulin Oz from the CMRR, University of Minnesota, and she will be talking about validation, reproducibility, repeatability in MRS.
Thank you very much. Thank you very much for having all of us here in your beautiful city of Lausanne. I was given the topic of validation, reproducibility, repeatability in MRS, which is quite broad. So I hope I do justice to it. So first of all, starting out from validation versus repeatability and reproducibility. Validation, as you probably realize, is a very broad term. It can be interpreted in many different ways. You can have a biomarker, MRS biomarker, that you validate for sensitivity to disease, progression, etc. You can have your pulse sequence and validate it to make sure that you're collecting the data from the intended region of interest, etc. But for the purposes of this talk, I'm only going to be focusing on validation of the concentrations that we estimate using MR spectroscopy. So from that perspective, the talk is in two parts and in two domains. One is about accuracy. This is validation and agreement with the ground truth, which is very challenging for us, and reproducibility and repeatability, which is related to precision. So as shown in this chart over here, so you can think about accuracy as agreement with the ground truth and precision as agreement with self, so to speak. And of course, we want to optimize both, but it's not always feasible. So first of all, validation, and I mentioned I will only focus on metabolite quantification and validation of accuracy of concentrations. And in the ideal case, we would take, for example, the brain tissue, extract the tissue using biochemical methods, do the measurements of the neurochemicals in that tissue, and then compare them to our in vivo measurements. Well, this is very challenging. It has been done, I believe, a few times, but it's regularly not available. The true concentrations are not available to us. So we have few alternatives, and we've been hearing about them since yesterday in multiple talks. So question for the trainees in the audience, not old folks like myself, but what are some of the options that are out there that can tell us if our measurements are accurate? Just yell it out. Well, biopsy, that's invasive. Yeah, you can do that for muscle, not for brain, right? Well, unless, yeah, if you're undergoing surgery, if the patient allows it, consents to it, and that has been done. You know, some epilepsy work has done that, but I'm looking for alternative methods. CRLB, we'll get to that, but that's, well, what I'm talking about is, oops, using simulated data where we do know the ground truth, right? You simulate the data, you know the concentration, so you have access to the ground truth. What else? Where? Phantom data, yeah. And then third, in vivo data, and that's in quotes, that's not true validation, but we do a lot of validation against other existing widely used software, what we call gold standard in big quotes. And I'm going to give one example. I picked one paper to represent examples of validation because they were doing all of these three. And that's the recent FSL MRS description paper from our third speaker in the session. What they did was they used simulated data that were generated for the MRS fitting challenge by Gosha and Dinesh and Roland. And they tested their new end-to-end spectroscopy analysis package. And I'm only showing the example of 3 major metabolites here. So they have access to 20 data sets. They know the true values, and the quality and conditions differ between these data sets. For example, if you look at the right-hand side here, SNR goes from 20 to 160, and that is known. So are the true concentrations, and you can see the deviations in the low SNR data from the true value are large, and they decline as the SNR increases. So this is a really nice method, and they have 2 algorithms that they test in the software. And then, of course, you can go ahead and look at all metabolites and plot out their the agreement of the concentrations that you determine with the known ground truth. Okay, so next, phantom. They have a nice phantom with known concentrations. Same idea, comparing the concentrations that they get with the new software versus the known concentrations. And you can plot out percentage difference, etc. And finally, they did a comparison using 3 different types of in vivo data sets where they validated their method against the widely used LC model software. And just by even glancing at the data, you can learn a lot. For example, they are using water rate referencing and total creatine referencing. And for total creatine, total choline, etc., they both are pretty, reliable, it seems. But you go to a difference from 7 Tesla to 3 Tesla, the creatine ratios seem to agree more because there are a lot of other factors that affect the quantification when you do the water referencing. You go to MRSI, there's a lot more variability. So even at first glance, you can gather a lot of information from this kind of analysis. So that's all I have on validation, on the very broad topic of validation. And I'll switch to repeatability, reproducibility. And I'd like to start out with definitions. What's the difference between these two concepts? And I'd like to refer you to this paper, the consensus in terminology paper by Roland Price et al. So repeatability is the variability in your measurements when the setting is the same. Same operator, identical conditions, etc. For example, if you have an individual in the scanner and you repeat the measurements without changing anything, that's repeatability. If you repeat the measurements in the same day, that could be considered repeatability too. But if you go to next week or month, etc., then conditions will likely change. And then you start talking about reproducibility. And that's measurements repeated under variability in measurements that are repeated under different settings. And the settings can be method, operator, scanner, site, magnetic field strength, etc. And why do we care about this? We care because we have multiple sources of variability, technical variability and biological variability that we typically want to measure. For example, you're administering a drug. Is that generating the expected change longitudinally in that metabolite? You can only measure that if your technical variability is lower than the biological variability that you're trying to measure. And I think a really good example of that is functional MRS, because the changes that are detected in response to stimuli are can be so minute that you have to be excellent. You have to have excellent repeatability to be able, or technical repeatability to be able to detect those. So I have an example here, a 7 Tesla study by Bednarik and Manja, and looking at the changes in metabolite levels in response to stimuli. Lactate, this is widely reported, widely understood. The percent changes are rather large, like 20%. But glutamate, we're talking about detecting 2 to 3% fluctuation. So your measurement precision has to be absolutely perfect with very high quality data like this one. So high within session repeatability allows the detection of these very small changes. Another example on repeatability where we looked at a conventional MRS protocol versus an advanced MRS protocol. So this is a standard steam press that comes on the Siemens scanner versus an optimized semi-laser protocol that includes the B1 calibrations and B0 shimming and everything. So single shot data acquisition. And data are collected from posterior cingulate cortex, and we compare the repeatability of these acquisitions by dividing up our data. This is a 5-minute data acquisition, 64 shots. We divided them up into 4 shots, 8 shots, etc., and determined the test retest CVs within that acquisition, comparing the conventional versus the advanced protocol. And these are some example metabolites where you can clearly see the lower repeatability, higher test retest CVs with the conventional protocol versus the advanced protocol. And these were 30 healthy elderly individuals, so not exactly a simple clinical cohort. So for testing physiological changes, like in the functional MRS case, or medication effects within session, you worry about within session test retest repeatability, which is easily quantified by the coefficient of variance, that is standard deviation of the measurements over the mean. And the rule of thumb is changes that are greater than twice the CV can be detected in individuals with 95% confidence. And Kramer-Rao lower bounds come into play here. So they can be a useful indicator of within session repeatability, but they are definitely not perfect. And I'm going to show an example of that. So they should be taken at guidance, but not the final word when you're designing a study and when you're trying to detect a change, physiological change within the same, even in the same session. So when you go to between sessions, like the typical drug trials utilize, then you have to worry about additional factors. You have to worry about hardware, software performance, subject placement, reproducibility, voxel placement. There are now methods to more reproducibly place the volume of interest based on anatomical markers and based on templates. And then, of course, you worry about day-to-day physiological variance. For drug studies, you may want to use same time of day or post dose interval would likely be critical. So these are the things that you need to keep in mind. Good news is you can get very reproducible data as long as all the technical factors are optimized and consistently applied to the protocol without changing parameters from, in the same subject from this week to next week, etc. This is from a study that we published a few years ago where healthy volunteers were scanned 4 times with weekly intervals. And these are data from 2 regions of interest at 3T and 7T. These are data that are overlaid all from one subject, the 4 data sets overlaid in each panel, the 4 weekly data sets. And both at 3T and 7T, you can see that as long as the protocol is optimum, all the B0, B1 calibrations are applied consistently, you can get highly reproducible spectral quality. Well, how about concentrations and Kramer-Rao lower bounds and test-retest CVs? So I don't have the concentrations here. The original paper has those. Let's look at the Kramer-Rao lower bounds on the top panels for the 2 regions of interest, posterior cingulate and cerebellum. A rather easy one and a bit more challenging region of interest. And comparing 3 Tesla versus 7 Tesla, you see significant improvement in Kramer-Rao lower bounds going to 7 Tesla from 3 Tesla. And you're like, all right, I'm happy. I have better repeatability. You look at the actual test-retest CVs, at least for the major, the highly concentrated metabolites, NA, creatine, choline, mannose, glutamate, there's no difference in the repeatability. So we already reached a limit of repeatability, best repeatability at 3 Tesla, and 3 Tesla is okay for those. You go to the other metabolites, more weakly represented metabolites, glutamate, glutathione, glutamine, etc. That's where you see, at least for these voxel sizes, that's where you see the benefits of the 7 Tesla ultra high field very clearly in the repeatability also, both in Kramer-Rao lower bounds and the test-retest coefficients of variance. Lower Kramer-Rao lower bounds do not always translate to lower test-retest CV. So these are things to keep in mind. You go to multi-site studies and things are even more challenging, multi-site reproducibility. So you have to worry about pulse sequence and parameters, how well they are matched across sites, all the calibrations and calibration procedures. And then finally, of course, the post-processing and spectral fitting. Centralized analyses will always be preferred in this case. Although if you take the same data set and analyze at one site and analyze it at another site, like we had done in one study, and you get the identical results, then that gives you a lot of peace of mind as well. So this is a figure from the first clinical consensus paper where we presented data from individual subjects, cerebellum, again, one of the dear regions of interest for my career and interests, and a challenging one at that. And with data acquired at Siemens 3 Tesla scanners at 3 different sites, different operators, different healthy volunteers, but using the identical protocol, sequence and protocol that was developed at CMRR and shared with the other site. And you look at the spectra and you can barely see a difference in the quality. The water, the residual water tells you these are truly different spectra. So data can be acquired highly reproducibly. And then we did a study with one of those sites, the Paris site, where each site acquired data from the vermis, the cerebellar worms and the pons, another highly challenging brain region, from healthy volunteers, 20, 30 volunteers at each site. First off, you see the spectral quality is very comparable. And then when you look at concentrations, these are the means and standard deviations in these different healthy cohorts. And they are really comparable, meaning data can be pulled between different sites. And the main driver for this kind of work in my career, at least, was a focus on rare diseases, where single site studies are very difficult. You need to be able to scan patients at multiple sites with the same protocol and pool those for example, potential clinical trial use. This is the next stage where we introduce multi-vendor reproducibility in addition to multi-site. And this was a 4-site study with 2 vendors, Siemens and Philips, done at 7 Tesla, with 7 healthy volunteers who were scanned and rescanned at all these 4 sites. And the additional considerations here are, of course, pulse sequence parameters, especially between the 2 sites. And you're going to learn in the next talk from Dinesh how this can be done very well. This is not the best example of that kind of harmonization. This is just an early example from our work. And then another important consideration is voxel-based calibrations, how they are done. This is for single voxel at each site and on each vendor. And of course, the post-processing and spectral fitting. And these are data, again, overlaid from one of those 7 participants. We collected data from the gray matter and from the white matter, and the test, retest acquisition at site 1, site 2, site 3, etc. And again, just by visual inspection of the data, you can see the SNR is very comparable, the quality is very comparable, and the spectral pattern is very comparable between different sites. And then we moved on to the quantification, and there we started to see some differences. For example, creatine between the 4 sites seemed quite comparable, but there are site-related biases in the NAA quantification, similarly on glutamine. Choline, very nice, comparable across sites. I will not go into details, but there were hardware-related differences and some acquisition-related differences, like shimming between these sites. And this is the last point I want to make from this work. When we look at concentrations obtained from data collected at scan 1 versus scan 2, we have this nice correlation. These are again, the major metabolites, the highest concentration metabolites. And if you look at the distribution, these are all healthy volunteers. So essentially, these data tell us that between subject differences in the healthy brain are detectable, at least for these most reliably quantified metabolites with our methods. Okay, just a summary of factors that affect reproducibility. Spectral quality, spectral quality, spectral quality. If you haven't gotten the message through all the talk, that's really, you want to start out with good quality data. voxel placement is really important, both between sessions and between sites, matching sequence parameters, and streamlined post-processing and spectral fitting, ideally in centralized analyses. So when you're designing a study, question is which variants do you want to evaluate? As usual, it depends on your question, depends on your study design. For example, if you're trying to detect within session stimulation effects or drug effects, physiological changes, you want to worry about repeatability. If you're looking into detecting treatment effects over days or weeks, you want to evaluate between session reproducibility using your method of data acquisition and analysis. And then if you're primarily interested in differences, group differences, for example, patient control differences, you need to evaluate within group variance, and remember that that variance may be different within your healthy volunteers and the patient group. They may move more, or they may have more variability in the concentrations that you detect. And these are my acknowledgements of all my colleagues, collaborators who have contributed to the examples that I presented from our lab. And I look forward to your questions.
Thank you very much, Gulin, for the very nice and clear presentation. So there is time for a couple of questions.
So I was interested by the sort of data quality. And I guess my question is, at what cost? So you would get higher quality data if you spent an hour preparing for a 5-minute scan, but you might also get better, higher signal to noise by running an hour of spectroscopy. So I suppose, obviously I agree that higher quality data is better, but what is a reasonable prep time for scanning? And at what point is the tipping point where we shouldn't try harder?
The good news is you don't have to spend an hour or even half an hour or even 15 minutes to prepare for the spectra that I presented. 3-4 minutes. And now we have a protocol, at least on the Siemens system, that does all those calibrations automatically. I mean, it's a poor man's version of automation on the Siemens system. The B0, B1 calibration, everything is queued. We have a video, maybe Dinesh will show, but okay, so you'll see it. Yeah, you go through it, you select your volume of interest, which is also automated now in our protocol. You select your region of interest from a drop-down menu, hit go, and then the rest is you sit back and relax and get the nice data. And we are already using it, this protocol, in several multi-site clinical studies and having good experiences with people who have never done spectroscopy before. So I'm thrilled about that.
Thank you very much.
Are there any other questions? Well, if not, we can thank again the speaker. Thank you. And the next speaker is I think we should move on, and we can have more questions for the next speaker, who is Dinesh Delchan, also from the CMRR, University of Minnesota, and he will talk about vendor harmonization in MRS.
Okay, can you hear me now? Yep. So, not moving. All right. So the International MRS Clinical Consensus Group, documented the clinical utility of proton spectroscopy in the CNS disorder, and they emphasize that there's a critical need to standardize advanced data acquisition and analysis in the clinical setting. This is in order to improve reproducibility of data quality and neurochemical quantification between site and vendors as shown in the previous presentation. So why do we need to, why reproducibility is important? So I just did the Google Press Spectra at 30 milliseconds, and that's what I came up with. So just if you look at the spectral quality, you can see some of them has lipid contamination, some is broad. So basically, and here I have data acquired from the same subject, but different protocols. And you can see it's very different. So like it's broader with a vendor provided compared with the advanced in the same subject. So if you look at the commonly used press sequence and free tester, so we have a 90-degree and 2 180s. So we can see on the free MR vendors, GE, Philips, and Siemens, the pulse pattern and duration are different. And I'm giving the numbers for duration there. So this is for a case when we use the body call for transmit and the head call for receive. So you can see there's differences between the duration and pattern. So this means that you'll have different chemical shift displacement if you run the press on different vendors. Also, it is known that if there's not enough power, the R4 duration will increase in the press sequence, I believe, on all vendors. And in addition, the interpulse delay, tau1 and tau2 are also differently implemented on different vendors. And this might affect the spectral pattern of J-coupled metabolites, such that if you are acquiring multi-site data using these sequences, you will need to simulate different basis sets for each vendors based on the delay and pulse. In addition, if you look at the water suppression and OVS, we can see that they are very different for the water suppression. Like we are using different water suppression technique. While OVS on G and Philips, I believe it's on, while on Siemens, by default, it's off. You will need to manually place the OVS around your voxel. So here's an example of data acquired using vapor and press. You can see if we're using the water level, water residual level is much higher in wet compared to vapor, and this affects the baseline of your spectrum. In addition, and this data I would mention that it's acquired on Siemens, where no OVS was used. In addition, you will get some lipid contamination. And here's the water to total creatine ratio. So you can see it's much higher water residual if you use wet. So what influences, so these differences will indeed influence the quantification of metabolites. So here's the example I showed previously. If we are using the same data acquired with a vendor provided press and our semi-laser, you can see there's differences in several metabolites, which means that it is challenging to pull together data from, if we are using data from multiple sites using the press sequence. So recently there was a paper by IMCL where they acquired data on the press sequence using from multi-vendor and site. So they tried to match the repetition time, and they match the repetition time, echo time, and voxel size, and they use a PCC voxel. And they did get a good spectral pattern between vendors, as shown here. I think it's a stack plot from all the subject at each site. However, when they did a linear mix effect model, they observed a higher contribution of site effects in the total variance of most metabolites. This was related to the difference in the methodology used between sites. And they suggested that in order to minimize this effect, we need to standardize the MRS protocol. So what do we standardize in the protocol. So first, we have to standardize the localization sequence, which include the water suppression, the obvious modules. So basically, we need to match the RF pulse, pattern, duration, the spoiler gradient, the interpulse delay, and the phase cycling. In addition, we need to do some pre-scan calibration also. We need to do voxel-based visual shame and B1 adjustment to have optimal SNO and good water suppression. So the methodological paper by Martin showed that the press has large chemical shift displacement at 3 Tesla and above. And it was recommended to use the semi-laser at 3T and above. So in this study, we're going to focus on the semi-laser sequence. So the semi-laser basically has 190 in one direction for size selection, one pair of 180 in second dimension, and another pair in the third dimension. So I've shown in the red diagram here. In addition, we have a water suppression module, and we have a 4 module of OVS. Since we are using the adiabatic pulses in the Y and Z direction, usually when you use adiabatic pulses, because of a good slice selection, you usually don't need water OVS in this direction. But here is showing that, I think it's this one. So we need to add the OVS module in the Y and Z in order to get rid of some artifact. So here I'm showing like in single shot, just one shot, we are getting artifact-free spectrum with this sequence. So also, as Gulin showed previously, we have high within and between site reproducibility at both 3 and 7 Tesla. And for the major metabolites like total NAD, total creatine, choline, glutamate, and inositol, the test-retest CV was below 5%. This one's already shown from last slide, last presentation. So one of the challenge when we faced when implementing when standardizing the semi-laser in the free vendors was how much B1 is available when using the standard body coil for transmit. So I think on GE, we have 20 to 25 microtesla on GE and Siemens. But on Philips, it depends on the coil configuration and transmit mode, and it varies between 13 to 22 microtesla. And I think the 22 microtesla is when you have advanced application like multiband. So since the majority of 3 Tesla uses a dual body transmit coil, where the B1 is around 15 microtesla, we set our constraint to 15 microtesla when designing our RF pulse. And just in the original S laser, we are using a HS1 or 25 pulse, which requires a B1 of 25 microtesla in the original S lasers paper. So I showed in the previous, in the 2016 MRS workshop, that we considered several Goya modulated pulsars, gradient modulated pulsar like BASI, foci, HS, and Goya worst. And we chose the Goya worst using a bandwidth of 10 kilohertz, HS modulation of 16, gradient modulation of 4. And if you look closely, the chemical shift displacement was only 1.3% per ppm. So we use this pulse and implemented it in the S laser sequence. And here's the amplitude modulation, gradient modulation, and the frequency sweep of this pulse. So the MZ profile of this pulse is compared to the original HS4 or 25, which is much sharper, as shown by the black plot here. Similarly, off resonance, you can see it's still, we have some smearing inside, but outside it's still pretty clean. So we did standardize S laser on the GE. So this one was done by Ralph Noske, on the Philips by Adam Barrington, and by me on the Siemens platform. And I think that's after you wrote the code and you get the simulation of a pulse sequence. That's what I got from all the different platforms. So you can see that we have a gradient modulation pulses, and all the gradients are matched, and everything is matched, like the ramping, everything is matched. So in addition to the localized sequence, we also standardize the vapor module. Since, as I mentioned before, the press uses different, so we had to optimize the vapor module. So we use the 8 water suppression module for human. And for the water suppression, we use a 30 millisecond SLR pulse, and with this interpulse delay. And the bandwidth for the pulse was 70 Hertz at full width of max. So for the OBS, we use a 5.12 millisecond HS1 pulse on GE and Siemens scanner, but on Philips, because of a limited B1, we choose to use an 8 millisecond HS1 pulse. And this gave comparable profile when used as saturation, as shown in the plot here. So in addition to the standardization of a similar laser with a vapor and OVS module, we also tried to do voxel-based B0 shimming. So this one shows the importance of doing both first and second order shim. So if you just do first order shim only, or if you do first and second, you can see you're getting an improvement of, I think, 5 to 7 Hertz in your line width. Yeah, you can see on the NA and minor stall here. So on Siemens, we use the 3D GRE shim or force map shim. So the 3D GRE shim is provided by Siemens. On Philips, we use the pencil beam 2D RF pulse, I believe. And on GE, I was surprised that when doing single voxel, they don't correct for the second order shim. So we work with Christoph Juhem using their Pharmaceter to adjust both the first and second order shim on the GE platform. So for the B1 calibration, uses the Bloch-Ziegler of resonance to get the B1 inside the voxel. On Philips, it was done over a slice using the vendor-provided power calibration. And on Siemens, it was just arraying the flip angle and determining the maximum of your water peak. So that was a final harmonized sequence across vendors. So we have a excitation pulse of 2.6 millisecond asymmetric sync pulse. The Goya was for refocusing 4.5 millisecond with this interpulse delay of 8, 12, and 30, 10, which give an echo time of 30 milliseconds. And we kept the same spectral width of 6 kilohertz between vendors with 2,000 points of complex point during acquisition. The ramping time and spoiler gradient was also matched. And when acquiring Metabolite, the center frequency was set at 2.67 across vendors. And while the water scan was done on the water peak without any centered change. So we use the G Discovery scanner at the University of Michigan. The Philips was at Johns Hopkins, and the Siemens, we had it at CMRO, University of Minnesota, and the Mayo Clinic in Rochester. So we use the body call for transmit. And for reception, we use a 32 Hz channel, head array coil. On G, it was a Nova coil, and Philips, it was a standard vendors provided coil. So here's the phantom data acquired from the brain of phantom. You can see that we got similar spectral pattern between the three vendors here. That's the SNR and line width we got after doing voxel-based B1 and B0 calibration. So this means that the performance is comparable between vendors and phantom. And also, we also acquired data from the 3 sites and in 5 different brain regions, as shown here. It was 5 subjects at each site and from 5 brain regions. And that's a stack plot of all subjects. So you can see we got comparable, highly reproducible and comparable spectral pattern between individual and between platform. So you will see on ponds here, there was one outlier where we could not get good water suppression. So that's why it's gray here. I think in ponds and hippocampus. So if you look at the SNR and waterline weight, waterline weight, you can see we got comparable waterline weight in all brain regions. So there was no statistical significance. While if we look at the SNR of NA, it was also comparable except for hippocampus and PCC. So you can see on Siemens scanner, the SNR was much higher in PCC and hippocampus compared to the other scanner. And we believe it might be related to how the head auricle was constructed. It might, because it's different between vendors. So that's the concentration we measured for these 5 neurochemicals. Here also it was comparable concentration, and also the Kramer-Rao lower bound was similar. So we also did study healthy controls and patient with SCF3 here in the bounds, in the cerebral white matter, sorry. So here we can see that the data is also comparable if we are acquiring data from healthy subject or from patients. So far I've been talking about the standardization of S-laser. So Richard Adams' group also did the standardized mega press sequence. So here also we are showing the Philips, Siemens, GE, and Canon, and they did a universal editing sequence where they match the 90-degree and 180-degree in the press sequence, and that's the information there for the duration bandwidth on all 4 platforms. And on Phantom, if you use the vendor-provided sequence for, let's say, GABA or lactate editing, you can see lots of differences on Phantom. And if you use the universal pulse, you can see the variation is much less if you use the same universal sequence. And they got good data on the GABA on the Philips and Siemens, and I think on the GE and the other scanner, Canon scanner also. So lastly, we went a step further, and we recently published this paper called Plug and Play Advanced MRS Protocol, where you will, let's run this. So basically you will, everything is automated. The only thing you have, the operator needs to do is select which voxel they want to acquire the data, and the B0 shim correction, the B1 correction, everything is done automatically. So you just need 1 click or 2 clicks, and that's it. And at the end, you will get all your data you need. So this is so far has been implemented on the Siemens scanner. So if anyone is interested to implement it on Philips or GE, I'll be happy to have. So the standardization of S-laser would not have been possible with collaboration from several sites, which I would like to thank here, and thank you for your attention.
Thank you very much, Nimesh, for the next presentation. So the floor is now open for discussion.
Very nice, Dinesh. Quick question. You indicated already there might be differences in hardware from particular several vendors. How do you best detect them, meaning distinguish them from protocol differences, and how could you best correct for them? Traveling volunteers, or what's the solution there?
I think if you acquire B1 maps for receive, you might be able to see the difference. But we did not acquire B1 maps in this study.
Thank you.
Thank you very much. This is really wonderful. I'm working also on pediatric patients or pediatric subjects. They have a low body weight. When I use a normal standard S laser, for instance, on a Philips scanner, my shortest echo time becomes 40 or 45 milliseconds. Do you know whether this is possible also with low body weight scans?
What do you mean low body weight?
Well, typically when the body weight is small, then your doses are different. So your echo time, your shortest echo time is increased, probably for SAR limitations.
I think we scanned on the free tester scanner, we did animal using the same sequence, and we were getting actually better echo time, like 26 or 28 milliseconds.
That sounds promising. I would love to check it. Thanks very much.
A very good presentation and amazing work. I will add two questions, in fact. The first one, have you encountered any kind of discrepancies between the manufacturers regarding SAR monitoring limitation regarding the semi-laser sequence? And second question, do you think you could extend this work at 7 Tesla with much more challenging situation relating to bandwidth, SAR, and clinical shift artifact?
Coming back to your SAR, no, we did not monitor the SAR during the study. So we don't know what the SAR between vendors is. And for 70, it might be difficult depending on your voxel location and if you're doing B1 stream or not. That's really depend on your voxel and B1 streaming, or if you're doing just one channel. Did it answer your question?
Well, I guess at this stage, if it's for multicentric with people who are not always experts, B1 streaming is not necessarily an option, I guess. So unless you manage to implement that everywhere, but
no,
wouldn't be CE approved anywhere else.
So on 70, we are using the NovaCall, and we have been sharing our protocol with different sites, and it's been working well so far on Siemens.
Thank you.
Yes, with the electric padding and the NovaCoil, one transmit NovaCoil. Thank you.
Are there any other comments or questions? If not, we can thank the speaker again. And I will leave the microphone to Jeff.
So the next speaker is Dr. William Clark from University of Oxford, UK. And he will be talking about denoising for MRS and MRSI.
Thank you. Thank you for the introduction and the kind invitation to speak. Is the microphone okay? Yep, great. I'm going to cover the topic of denoising in MRS and MRSI, which I feel has already generated a little bit of discussion here, so I look forward to the questions. So I'm going to give a very brief summary, I hope, and then an extremely brief coverage of what's in the field, but then dive a bit deeper into one of the techniques, which is low-rank denoising. So let's get going with some definitions, starting with a signal model for MRS, where we have some observed signal, and then some white Gaussian noise, which contaminates it. And the noise is thermal noise, which in human subjects arises from within the subject, whilst in smaller samples, like rodents, it predominantly arises in the coil and the electronics. So if we have this simple signal plus noise model, we can imagine a denoising operation which sort of reduces the variance of the noise component and then outputting a higher SNR spectrum without spending any more experimental acquisition time on it or affecting the underlying signal component. So with that, we kind of have a denoising holy grail where we aim to reduce the influence of noise, preserve all of the underlying signal variance, and hopefully as a bonus, though it isn't essential, does so in a way which doesn't need us to radically redefine our analysis techniques. But I'll quickly say that that this signal model is obviously incomplete. As we've heard several times over the last 2 days, artifact also contaminates the observed signal, and some of that appears like noise, and some is unwanted and unexpected signal infiltration. And I would say that dealing with this artifact is in some ways far trickier and possibly more important than dealing with noise in MRS. And why is this? Because artifact ruins our most successful analysis tool, the highly effective denoising that we use in every single analysis, which we call fitting. So by using extremely accurate simulations, which you might use Mars for, through fitting, we can fit an accurate model to our signal component, thereby substantially and almost entirely mitigating the effect of noise on our analysis. If the fitted model is accurate, i.e. there aren't artifacts and bias isn't introduced, then this should definitely outperform any data-driven denoising approach. But so effectively what I've said is that you shouldn't want to denoise and fit in the same time. And why is that? Because when we fit a model to denoise, we reduce our statistical degrees of freedom. So taking a really simple example, a popular approach for modeling data is a low rank approximation. And if we have a dense full rank matrix, so with an A by A matrix with 64 degrees of freedom, and we truncate it to a rank 3 approximation applying this model, then we almost halve the degrees of freedom. Those 64 numbers in one matrix are described by just 39 numbers in this way. But by doing this, we kind of necessarily bias our approximation of the dense noisy data. And we're also going to cause correlation between entries. So then any subsequent attempt to carry out least squares fitting after denoising is going to violate the conditions on that least squares fitting and result in overly optimistic uncertainty in a biased answer. And so fitting multiple models in series to the same data, for example, denoising and a spectral fit or denoising plus statistical tests, is not going to bring any additional benefit. So that kind of leaves the questions where can we apply denoising? And so we can apply it kind of anywhere we're not going to do any statistical inference. And so in a normal trial, maybe we're going to try and perform our primary statistical test between subjects or groups of subjects. And as I've said, we're going to carry out our sort of excellent denoising operation in the time or spectral domain through fitting. But we also often collect a spatial grid of spectra where we generally, not always, but generally don't make any comparisons between the closest voxels. And also in this grid, we expect metabolite signal to be varying quite smoothly or varying across a low number of tissue compartments. So we can exploit that to infer a less noisy representation of the signal in a data-driven way. And I should say that this might also work for other sort of time courses of spectra or other dynamic spectroscopy applications. And I'll just cut to the chase by saying that denoising across the spatial grid works. And this is very obvious from visual inspection. The results here are from some 3T phosphorus brain spectroscopy at quite in 8 minutes. That's an application famed for its high SNR and high resolution. And I've denoised it and I've compared it with some data which I spent 45 minutes collecting. And so you can see visually there's a big increase in apparent SNR, and then it compares quite favorably with that 45-minute data looking quite similar. So qualitatively that works, and that's backed up by quantitative interscan reproducibility, which is also increased. So here you see the interscan reproducibility for 3 metabolites, or sets of metabolites, calculated from 10 repeats on each of 6 subjects. And so where denoising has been applied using the spatial information on the right-hand side of the screen on this side, you can see an improvement in reproducibility here shown as a standard deviation. So we have a lower standard deviation. But where we don't use the spatial information and only try and apply denoising before fitting using the spectral information, it actually reduces the reproducibility, it increases the standard deviation. But I should say that denoising doesn't actually work as well as it seems by initial visual inspection. The apparent SNR is not a good measure of denoising performance, and that's probably the most important message today from my talk is, yeah, don't trust what it looks like. You have to go deeper. And that's seen in the comparison on the left, where the fit estimated uncertainty, so what comes out of the Kramer-Allow bounds, is a really poor estimate of the true uncertainty here, estimated using a Monte Carlo procedure, except for in the original noisy data, which I haven't applied denoising to. And in fact, it gets a bit more complicated. The performance is metabolite specific. And so one has to be very cautious when applying and interpreting the results. But more on how we can do that later. Now I want to have a quick run through of some techniques which are out there. And this is a very quick run through. So apologies if I missed things. So in the literature, sparsifying and smoothing techniques of spectral time series was really nicely summarized by Ben Rowland in a comprehensive analysis using some time-resolved phosphorus MRS. And in one case shown here, they use SIFT, Fourier thresholding technique to remove low variance frequencies from the spectral domain. So by Fourier transforming the time series of spectra, you then threshold, shown here, and then convert it back. And the result then is a visually denoised time series. But in comparison with simple windowing, either with or without a Gaussian kernel and spline fitting, the advanced sparsifying methods, including wavelet transforms and so on, came out very much second best to that simple windowing procedure. So denoising can be effectively carried out using very simple methods, using smoothly varying signal. Another major group is the low rank methods, and recently there's been a kind of flurry of interest in this technique, with really nice examples of work by Jesse Mosso for diffusion weighted MRS and Lauren Rum at RSM for phosphorus MRSI. And here we see the LoRa approach as a low rank approximations method developed by Neugen et al. As applied to MRSI data, but lots more on low rank approaches at the moment. And in fact, this slide was preempted yesterday by sort of saying that, yes, you can also include denoising in a reconstruction step. And this produces very high SNR and high resolution MRSI, but signal is limited to a kind of linear combination of what's constructed by the small range of components left after denoising. Okay, so I did promise it was very, very truncated review of methods, and that was it. But that's so I can spend a bit of time going over what low rank denoising is and sort of how it's applied and stepping you through the process. And it's very popular in MRI at the moment, where Marchenko Pasteur and the related Nordic techniques are kind of being pushed for diffusion and functional imaging. Again, situations where you repeatedly acquire images. So our first question is kind of, can we use low rank for MRSI data? It's a good point to start. And so what I've done here is set up a little toy example where I've got a 1D series of voxels, and there's 3 peaks, and each has maxima at different positions. And it's represented in 2 ways here, kind of with spectra pulled out, and then everything concatenated into a single matrix here. And you can take that matrix and carry out a singular value decomposition and SVD, that's otherwise kind of known as a PCA, to represent it as a set of left and right singular vectors and related singular values. And what I'm showing you here is just the non-zero, singular vectors related to the non-zero singular values. So you can see they decompose into some spectral components, the spatial components, and there are a bunch more zero value singular values. And this data can actually be equivalently be expressed as a sum of 3 rank 1 components. So the product of each pair of vectors, each scaled by the related singular value. And we see that again here. And if you sum those together, having scaled them, you return our original data. And that's sort of the setup. But you're all saying this is noiseless data. What's it doing in a denoising talk? Well, we can add some noise to the data. And of course, we see some noisy spectra, and we see our concentration estimates get worse. And what we see after we've done the SVD is some more non-zero singular values corresponding to more rank 1 components. But these components sort of seem to contain mostly noise as compared to the 3 kind of signal components. So I've got a red cursor on a red thing. So these 3 signally looking components and a noise one, and a bunch more corresponding to these single values. But if I zoom in on this graph and change the contrast a bit, we can see that, in fact, our signal components also contain some noise-like structure, and these singular values have, in fact, changed as well. And this tells us there's no such thing as a pure signal component, and conversely, there quite likely to be some signals, some more variance residing in our noise components. So how can we actually denoise using this? Well, it's very simple. We just zero out all of these singular values that we think are mostly noise, i.e. dropping those components. We do that, we've denoised. And if I go backwards and forwards, we can see that the spectra have indeed been denoised and our concentration estimates get a bit better. So in that case, selecting the point at which to cut off our components was very easy. I had set up an explicitly ranked 3 problem and then chosen 3 ranks to keep. But how can we do that in the real world? Well, we can do manual inspection, and it might be as simple as looking at our result and picking a kind of point on the graph where it becomes obvious. But that gets very slow if you have lots of spectra, or sorry, lots of subjects, or you're applying it in a sort of sliding windowed approach. Another way we can do it is we can know the underlying physical equation. So Cole combination, we have Rommer's equation, which tells us it's an explicitly rank 1 sort of solution. It's just a linear combination of scaled coils, and that's what procedures like WSVD does. Or we can use data-driven methods. So one is the Marchenko-Pasteur method, which uses statistical estimation of pure noise singular values to set a limit on the singular value magnitude. So you'll see histograms like this. This is for the data I've just shown, where low value singular values are seen, and the procedure tells you a limit at which to draw a line and exclude below. Another is Stein's unbiased rank estimate, otherwise known as SURE, and that actually provides an analytical solution to the relative mean squared error of truncation to each rank. And so that's another approach, and in this case, actually, everything gives the same answer. But we can do some other things. Rather than just zeroing or keeping components, we can use a different approach where we, in fact, scale the components we keep. In fact, we can do anything we like with our singular values, but only some things make sense. So you've got a hard thresholding technique, that's what I've been talking so far, or all the ones that you keep above a threshold, you can scale down, sort of weighting the more higher value ones more than the lower values. It's actually been proven that this approach is mean square error optimal, which readily becomes apparent as we take our simulated data and increase the noise. So it's the same data as the last few slides. As we increase the noise, we can see a separation where the sort of the optimal soft thresholding becomes gives you a better result. But there's a massive caveat with techniques which provide mean square error optimality, and that's the fact that the mean square error of the data, i.e. the spectral data, is not equivalent to the mean square error of the estimated parameters, i.e. a lower mean square error of denoised spectra related to the kind of noiseless spectra doesn't give better metabolite estimates. And that can be seen here in this slightly confusing graph where different methods are compared. Don't worry what the methods are, but clearly you can see that looking at the spectral RMSE versus the fitted concentration RMSE, there's a different pattern here and a different order, and it changes across noise levels. And so, for example, that optimal soft thresholding is always minimum. In this case, in the spectral RMSE, it's mathematically guaranteed. But if we look at its performance with concentration estimates, it's terrible until we get to high noise, because it's better at dealing with mixed noise plus signal components. And so there's a trade-off there on depending on noise level and what method you might want to use. So yeah, that's the purple line there. One of the tricky issues to deal with as well with low rank denoising is that it introduces signal dependent variance. And this can be, you can prove this to yourself by carrying out a simple Monte Carlo simulation of the procedure. And as a result, it means that the true noise variance can no longer be, well, not noise variance, but variance across sort of repeated acquisitions can no longer be estimated from baseline regions. And however, there is an analytical solution to this recently published by Chen and colleagues in Pinas, And it uses the left and right singular vectors and the sort of original noise variance to calculate what the variance of each entry in your denoise matrix is. And this does a pretty good job of estimating the uncertainty in our denoise FID. So compared to the original noisy variance of a FID, we might guess that our baseline noise is our true variance, but actually carry out Monte Carlo estimation. This gray line shows that it's not that original that variance. And this method does actually do a good job of estimating it. So we do have a way of kind of forward propagating. I'll finish up by mentioning two important but difficult to understand factors. And the first is often denoising is applied in some sort of sliding window approach as opposed to on all the data simultaneously. And the second is that it might be important to center or demean your data before performing your SVD, kind of as people do with the PCA analysis. And it seems quite complicated to figure out the exact sort of results of those procedures, but the kind of the rule of thumb is it seems to reduce the effectiveness of overall denoising, but it dramatically reduces the signal-dependent variance to a point where the residual noise variance once again appears uniform and independent. So less performance, but much easier to use. It seems like a sensible thing to do. So it's clear in my eyes that denoising does have a role to play, but that extreme care must be taken to ensure that the methods applied actually improve the variance or reproducibility of something of the parameters we care about, which is the end concentration values, not necessarily how pretty our spectrum looks. And we also need to better explore how to optimize parameter selection and methods based on the effect of on those concentration parameters. Different methods sort of can produce quite different sort of results. So here we see some errors, and Marchenko-Pasteur method, for instance, in this case, does a very good job in some voxels, but a very poor job in others, whilst another approach does denoise reasonably, like to a lesser extent, but more uniformly. And it's going to take some quite advanced mathematical methods probably to sort of like message passing algorithms for us to be able to understand how choices we make affect the sort of uncertainty through the highly nonlinear fitting algorithm. And with that, that's me done. Thank you for listening, and especially thank you to my colleague Mark Chu, who was my partner in crime doing all this work on denoising. Thank you.
Thank you very much for this great talk. Do we have questions from the audience?
I'm wondering exactly when we should use this methodology of denoising. For instance, if I'm interested in a small peak in the spectrum that's about the same size as the noise, is this going to help me see that small peak and be sure that particular chemical is there? So in a way, I'm asking also, is this a linear operation? Because we're very concerned with spectroscopy that we have a linear vertical scale.
So the linear, I'll answer that in reverse. Linear, yes, if you use a hard thresholding. Soft thresholding, that's one of the difficulties of it with the scaling. Will you see that small peak? It depends whether the small peak is there present in multiple spectra. If it was there in only one and your noise was on the same order, it looks like noise, so it's very likely you'll remove it. If it's present in multiple spectra, then it should be picked up. And there is a choice on where you draw that sort of the line of truncation.
So I should only use this if I have multiple spectra. So if I have one spectrum, I can't.
Well, if you're going to fit it, fitting with a known model, which is highly accurate, is going to be better than any denoising, data-driven denoising, which doesn't have all that prior information, prior knowledge that you're leveraging.
I just had one other comment about the source of noise in a spectrum, and I think it's a common misconception that we're all transmitting RF noise. We're not. It's when you put a body inside an RF coil, it changes the electrical properties of the coil, and that's why the noise changes from an empty coil to a coil with a weakly conducting solution inside of it.
Thanks. Wonderful talk. My question is, I think you're the perfect person to ask because you know a lot about those things. When you would have time series data like functional MRS, or you want to measure T2 with multiple echo times, do you think it's better to denoise and then fit and then do your quantification, or do you think it's better to just fit already the spectral and the time course at the same time?
So
and you shouldn't do both, if I understand correctly.
I'm biased to say you should model both things at the same time. With the denoising, it's tricky. I think Jesse's actually done more work on the sort of across dynamic sort of work, and I think a similarly sort of complicated answer has come out. In that, in some cases, yes, in some cases, no. There might be other areas that you could use it. So it might be possible, for instance, to use it to initialize your fits in a much sort of more robust way by using the denoised data and sort of ignoring the sort of the messier sort of variance issues. So personally, I feel that probably I stick with my message from earlier on that you shouldn't use it where you're going to carry out, either do a second run of modeling or carry out statistical inference. But that's not to say denoising in those cases has no use, but it's going to be in a sort of secondary role, say in initialization or learning what's in your spectra first before you then apply a model. Yeah, that would be my feeling.
Okay, thanks.
You mentioned something about like if you make for MRSI that you could do the low-rank denoising either during reconstruction or separately from the reconstruction, and I did not get your kind of, is there like a conclusion or a recommendation that you can count that you have at the moment? Both ways are fine, or is there one that is more preferable than the other? Or
I would say that I haven't carried out the comparison. I don't know. Certainly the denoising after reconstruction is much easier to apply, and you can do it without the raw data. I would hesitate on giving you an answer without doing the comparison. There are so many choices to make in the reconstruction sort of route.
And as a part of the denoising or making sure that, okay, what you get as a signal, we are actually interested in the signal. What if I mean, there are methods that can calculate the entropy of the signal or of the, is there something like that, that done in these approaches that you look at the entropy of your signal before, I mean, your data before and after, and try to use also that as in addition to denoising as a.
It's a good thought. I haven't seen any work. Anyone correct me?
Something that Shannon did with information theory, like when they had to solve this at Bell Lab.
Mm-hmm.
I was looking forward for this talk. You showed before that denoising could enhance or improve reproducibility.
Yes.
So what about, and that is shown by reducing standard deviation?
Yes.
Do you ever go beyond Kramer-Rao lower bound, or in other words, is still Kramer-Rao lower bound a good measure for?
So those weren't Kramer-Rao lower bounds. There was a standard deviation on the metabolite concentrations. So as in the actual measured, we measured 10 times each subject in the same scanner run, and then calculated the variance over each voxel's concentrations.
And was that below Kramer-Rao lower bound from estimated from 1?
I can't remember, but the issue is the Kramer-Rao lower bounds you get out from the fitting is then invalid after you've denoised.
If I have time for another question, have you ever measured any bias or misaccuracy after denoising?
Yeah, there is plenty of bias in denoising. Here is some RMSE errors after denoising in basically exactly the same data I've been showing. Yes, there is. You can see that the the bias, there is obviously bias in the noisy data, but there is also bias in the denoise data, and it's different. Whether one's better than the other is up to you to decide.
Do you have one last question? If no, let's thank the speaker again for a great discussion. So our next speaker is gonna be online, and it's Yeong Jin Kim from Seoul National University in South Korea. I hope I didn't mispronounce your name. I'm sorry.
Okay, can you hear my voice?
Yes.
Okay. Thank you so much for introduction. And also I thank the organizing committee for this wonderful opportunity. First of all, I'm very sorry that I was not able to make the trip to Lausanne this time because of my personal reason. However, I do hope that my talk can be delivered to you as clearly as I anticipate. Today I'd like to talk about machine learning in MRS and MRSI as an MR spectroscopist. I'm not the expert on deep learning, of course, so I'm just gonna share my experience as an MR spectroscopist. Okay, before I start my talk, I would like to briefly go over the definition of machine learning and deep learning. Machine learning is an algorithm that enables computers to perform tasks without explicit programming. And deep learning is a part of the machine learning. However, in deep learning, we use structured algorithms called artificial neural networks. So in machine learning, first of all, we need to extract the feature, and then using those features, we train our classifiers. And however, in deep learning, those features can be extracted automatically by performing convolutions using convolution filters. And as we go deeper and deeper, the higher levels of features are available, which could be advantageous. So as we all know, the deep learning is changing the way we live in these days very quickly. And in MRI, deep learning-based reconstruction is about to be used routinely. And some of the vendors obtained already the FDA approval. Then how about in our society? In fact, the machine learning has long been used in our society as well, particularly on the quality management and pattern recognition. And however, these days, the applications of deep learning are increasing more and more, as you can see here. So motivated by these previous deep learning studies, we also conducted a couple of deep learning studies in MRS. And from the very preliminary experience, I would like to go over the important elements and the associated challenges in the development of neural network. And because of the limited time, I would like to especially focus on the quality management and quantification tasks. Okay, first let's talk about the types of input and output. These are some of the studies already published. And these two studies were for artifact detection. So in this sort of a classification study, the output of the network is sort of fixed to be the probability of classes. However, for input, we still have a lot of choices, in fact. For example, this study used a real and imaginary spectrogram, which is a representation of a two-dimensional representation of frequency and time. But this study just simply used the real spectra as their input. And these 3 studies were for quantification. And these 2 studies use amplitude or the concentration of metabolites as their output. However, in this study, which was our study, the task was to map the brain spectrum to a metabolite only spectrum. So as you can see here, even for similar tasks, we have different choices in the input and output. And one thing to note here is that all these approaches require pre-processing, such as Fourier transform and phase frequency correction stuff. So somebody among you may want to directly use the FID, the raw data, as their input, as your input for, let's say, full automation, including pre-processing. However, even in this case, we have 2 choices. First, we can use a real and imaginary FID in 2 channels as our input, or we could use complex FID in a single channel. And the complex FID in single channel is known to preserve the phase information. So this one may be preferred. However, in this case, we need a so-called complex network that is capable of complex convolution. And the FID is time series data. So that means a recurrent neural network may be more suitable than the convolutional neural network. So as you can see here, the choice of input and output can influence the rest of our study design, including the selection of networks. So now let's talk about the data preparation. Now you select your input, the type of your input and output. And so as a next step, we want to prepare our training data. And this data was taken from our recent study. And the task was a quantification. And as you can see from the slide, that as we increase the amount of training data, the quantification error tends to be lowered and lowered. So the more the training data we have, the better, of course. However, as we all know, in general, it is quite difficult to obtain the big in vivo data sets for training. So at some point, we need to simulate our data. However, realistic simulation seems to be, so far, according to our experience, the realistic simulation seems to be very challenging because of the sensitivity of neural networks. For example, let's see, in this example, the task was to restore a truncated FID to fully acquired FID. For example, let's say we have a truncated FID that contains only the first 64 points out of, say, 1,024 points. And we want to train this guy to recover the truncated FID to a fully acquired FID. And we trained it and we tested our network on the simulated data set first. And we obtained the FQN of 1.28. I mean, the FQN between the ground truth fully acquired data and the restored truncated FID. So the FQN was close to 1, which is pretty good. However, when we use our network on the true in vivo data set, our FQN is suddenly increased this way. So for some unknown reason, we don't know why exactly. So there's definitely something that the network does not like about the in vivo data compared to the simulated data. So it's challenging, and for better generalization, currently we are working on incorporating the so-called baseline variation into our simulation, which was not considered yet. So once you prepare your data, next step may be choosing your network. However, the choice of neural network is also very challenging because there's no theory on it. So including ourselves, people tended to employ the most recent one or the most widely used network, like AlexNet, for example. However, but as you can see here, that according to this very famous book, there is a optimal capacity or optimal complexity of your network. And if your network is more complicated than what it is required, and we are subject to more generalization error. So simply employing those already published or reported network may not always be a good choice. For example, in this study where the task was to predict the brain age from T1 weighted MRI images, where that the authors used a relatively very simple network, and yet they won the competition. So let's say we have chosen our network, the architecture. But like I said, just in the previous slide, we might need to design our own network, which means we need to optimize our network, our customized network. But as you can see here, even for this relatively simple network, we have many hyperparameters to optimize. So to optimize these hyperparameters at the same time, so-called grid search is one option. However, the so-called Bayesian optimization is known to be far more efficient. Bayesian optimization uses the Bayes rule to progressively optimize those hyperparameters. And it is known to be working pretty good according to these previous studies. And although the theory behind the Bayesian optimization may be quite complicated, however, we can simply use those open source, which is available in different deep learning frameworks, such as MATLAB and PyTorch. So let's say we optimize our network. So we are almost ready to test our network. However, at this stage, we should be very careful with out of distribution data. So by definition, the out of distribution data refers to all data outside the training range data. So, and importantly, networks are subject to unpredictable results on out of distribution data. So we want to definitely filter out those out of distribution data before we use our network. For example, in this example, the task was again the mapping brain spectra to a metabolite only spectrum. And we trained our network, but not including this sort of spectra, which is contaminated, severely contaminated by artifact or containing the abnormal level of metabolite concentrations. So these sort of data can be considered as a out of distribution data. And as you can see here, we have a substantial error. So we definitely want to filter this out before we use our network on practical data set. How do we do that? However, because according to the definition of out of distribution data, the range of out of distribution data could be unlimited. To solve this problem, we may want to develop another network in addition to the main network to filter out any out of distribution data. According to these previous studies that clearly demonstrated the potential of deep learning in quality management. In these studies, the task was to detect spectra with the artifact, and they reached very high accuracy even on the true in vivo data. So based on this approach, we want to develop our another network. So one potential approach may be using so-called NOGAN, which uses generative adversarial networks for anomaly detection. So in this approach, we use GAN, and GAN can learn how to synthesize the data that are very similar to the training data set from the random noise. And in this approach, we train our GAN using those data that we used in the training of the main network, the network A. And during training, the GAN learns the noise representation of the training data. So forming the so-called manifold of training data like this. So let's say we finish the training, and now we want to check, for example, we want to check this spectrum, whether or not this spectrum is within the training range or out of distribution data. And how we do that? We do so-called latent space mapping or back projection of this spectrum into the noise. And we do this over and over again until we find the optimal noise vector. And if it's really optimal, then again, using this optimal vector, generate a spectrum that is very close to the input spectrum, but except without the abnormality. So in this example, the noise level. So this generated data, that the spectrum can be considered as a, excuse me, considered as a normal version of an abnormal spectrum. So by subtracting this guy from the input data, we can nicely isolate the abnormality here. So in principle, any type of abnormality can be detected using this approach without explicitly training. However, the accuracy of latent space mapping is, to the best of my knowledge, is still remaining issue. So if there's anyone who would like to implement this sort of approach, they need to record this limitation with this approach. So let's say now we are completely rejected all those out of distribution data. But even after that, even on the training range data, our network are still subject to errors. In this case, a quantification error, but without providing any uncertainty information. So let's say, for example, this is the network we trained for quantification, and this guy nicely gave us the array of concentration of those metabolites, but that's it. You know, that's it. They don't provide any information regarding the uncertainty, even though we know that they are not perfect. So we definitely need to need some method to evaluate the uncertainty in the network output, such as the Kramer-Rao bound in the nonlinear least square fitting approach. So one potential approach may be the Monte Carlo dropout approach, which was developed from Bayesian deep learning approach. In this approach, we look for a distribution of network weights rather than a single set of optimized weights. And combined with the data uncertainty, such as a intrinsic noise in the input data, what we get is that we get a distribution of output instead of a single value to output. And from this distribution, we may be able to extract some uncertainty information. However, this process is known to be extremely complicated mathematically. However, fortunately, thanks to the recent work by Dr. Gar, we can relatively simply extract the uncertainty information by training our CNN with the so-called dropout operation. The dropout operation randomly sets some of the network output to 0 according to for the given dropout probability. So we implement this approach for the quantification task, and we trained our network. Again, the task was to mapping the brain spectrum to a metabolite only spectrum. But however, in this approach, we do inference multiple times, like 50 times. And at the end of the day, we get the mean and the variance spectrum. And from which we can obtain this sort of uncertainty-related spectrum. However, this approach is just one possible method. And to the best of my knowledge, there are different approaches being continuously reported. And these, for example, these two studies were reported at the ISMRM this year, and especially this study was an MRS study. Finally, we need to talk about the transparency. And it is well known that the deep learning approach has a severe problem of a black box. So and neural networks is known to be very easily fooled, And therefore, at the end of the day, we definitely need to check what they really learned, or what based on what they made such a decision, which is, however, quite challenging. But there are, at the same time, several techniques, such as a GradCam, that can visualize the most important region that was used by CNN in its decision making. I think this study is one of the best example that emphasize how important it is to check what CNN really learned. In this study, the task was to predict the functional outcome of a stroke patient from the apparent diffusion coefficient maps. And they reached very high accuracy, and they were so happy. But when they checked what they've learned, what the CNN really learned using GradCam, they surprised to found that the highlighted region, or the most important region, was not found in the infarct region, but found near the boundary. And they concluded that our network predicted based on movement artifact, not based on the degree or size of infarct. This previous MRSI study already used GradCam. And in this example, this spectrum was rejected because of the poor spectral quality. And this figure shows that this region is the one that influenced the most in the decision making by CNN. So as we know that we have a lot of different challenges, some of them really difficult. But at this point, nonetheless, the one clear potential advantage with deep learning technique may be rapid processing. For example, in this study, the author reported that it took only 2 minutes to conduct a quality management on 10,000 spectra, using the regular CPU. And in this study, they said they reported like it took 20 seconds to quantify 3 metabolites from 10,000 spectra. And if you use GPU, and you can speed up even further. So this rapid processing capability by deep learning should be definitely beneficial, particularly for 3D MRSI. I would like to summarize my talk. So according to the recent trend regarding the deep learning study, I think there should be more study coming up. However, given at the same time, given the variety of different challenges, at this point, deep learning aided MRS may be more suitable goal than deep learning based study. And some of the techniques developed for deep learning aided MRS may potentially further facilitate the clinical applicability of the MRS and MRSI. Thank you for your attention.
Thank you very much for the great presentation. We will open the questions. And thank you also for playing the cow game with us.
Hi, this is Roland Price. Very nice talk. Thank you a lot. I was particularly intrigued by your outlier detection algorithm. I'd not been aware of that too much. You showed it sort of, you can even use it to find the cause for why it is an outlier there. I was wondering, in our experience, outliers certainly, I mean, if it has not been seen by training, obviously it can't find things. In our experience, it seems also data at the edge of the training data, so you have a uniform range of parameters, and whatever's at the edge is not well estimated. Can that be used in that case as well?
Yeah, that's one of the limitations. And I also read your recent abstract, and I definitely agree with your opinion that deep learning the network supposed to show the its capability quite evenly across the data distribution. However, as you show in your in your abstract. And as I also seen during our study, and it's sort of biased. So as the data get close to the edge, there's a bias. So and nearby the boundary, and this technique may suffer some incorrect performance. So that's the definitely I agree with your point.
Hi, I have two questions, or a question and a comment. So the first one is for this anomaly detection, have you looked into diffusion models instead of GANs? Because diffusion models seem to be kind of GANs, but way better.
So unfortunately, I don't know about the diffusion stuff, but I'll check it out because
yeah, it's very new, but you know, it seems to be just doing better than GAN for pretty much everything. So it's probably better for this too. It doesn't require just this massive amount of training data that GANs need, which is kind of their major limitation.
Okay.
And the other comment I had was, you said for uncertainty, which is obviously very important for everything, just MRS, but really anything you use deep learning for. You use like a dropout layer or an ensemble. I think these techniques, you know, don't really have any statistical guarantees that they converge to standard deviations. There are other methods that do have these kind of guarantees. So there's an abstract in the ICLR conference from this year.
Okay.
I think first author, I know there's a middle author named Michael Jordan, and they have
Michael Jordan, okay.
Yeah. So and they go through kind of 4 techniques that you can use to basically for any model extract standard deviations, or something that at least will converge to standard deviation under certain conditions. So probably just something to look into.
Okay, yeah, thank you for your comment. I'll definitely check it out. Yeah.
Do we have more questions? If no, let's thank the speaker again. And this ends the session. So now the planning is to go to see the poster session, just there. And do we have special recommendation for after the poster session? So we meet there at or we go together?